ಇರ್ಬೇಕಾದ್ರೆ ಬೇರೆ ಎಲ್ಲ ಪಾರ್ಟಿಸಿಪೆಂಟ್ ಮ್ಯೂಟ್ ಮಾಡ್ಬಿಡ್ರಿ.
Yes, sir. So we can start it, sir.
Manjunath sir.
So I think his video is paused, sir.
No, I feel he is not connected properly. ಒಂದು ಸ್ವಲ್ಪ ನೆಟ್ವರ್ಕ್ ಚೆನ್ನಾಗಿಲ್ಲ ಕಾಣಿಸುತ್ತೆ. ಮಲ್ಲಿನಾಥ್ ಸರ್.
Yes.
ಮಲ್ಲಿನಾಥ್ ಸರ್ ಕೇಳ್ತಾ ಇದ್ದೀರಾ?
ನಾಳೆಗೆ ಬೇಕಿತ್ತು, ನಾಳೆಗೆ
ಮಲ್ಲಿನಾಥ್ ಸರ್.
ಅಲ್ಲೊಂದು ಬೇರೆ ಕೀಬೋರ್ಡ್ ನೋಡ್ರಿ, ಇದು ಅಂತ ಬೇರೆ ನೋಡ್ರಿ. ಒಂದ್ ದಿವ್ಸಕ್ಕೆ ಹೋಯ್ತೀನಿ, ನಾಳೆಗೆ ಒಂದ್ ದಿವಸ ಅಟೆಂಡ್ ಆಗಿ ಹಾಲಿಡೇ ದಿವಸ ಮತ್ತೆ 29 ಆಮೇಲೆ ಮಾಡಟ್ರಿ. ಸದ್ಯ ಬೇರೆ ಕೀಬೋರ್ಡ್ ಹಚ್ಚಿ
ಶಶಿಕಿರಣ್
ಸರ್. ಮಲ್ಲಿನಾಥ್ ಸರ್ ಕೇಳ್ತಾ ಇದೆಯಾ?
ಹಾ, ಕೇಳಿಸ್ತಾ ಇದೆ ಡಾಕ್ಟರ್.
ಹಾ, ಈಗ ಸರಿಯಾಗಿ ಕೇಳ್ತಾ ಇದೆ. ಆಗಲೇ ನಿಮ್ಮ ವಾಯ್ಸ್ ಬ್ರೇಕ್ ಆಗ್ತಿತ್ತು. ನಾನು ಬಹಳಷ್ಟು ನಿಮ್ಮ ಹೆಸರು ಕರಿತಾ ಇದ್ದೆ.
ಅದು ನೆಟ್ವರ್ಕ್
ಅದೇ ಹೇಳ್ತಿದ್ದೆ, ನೆಟ್ವರ್ಕ್ ಈಗ ಸರಿಯಾಗಿದ್ದಲ್ಲಿ ಕೂಡ್ರಿ. ಈಗ ನೀವೇ ಪೂರಾ ಇದು ಮಾಡ್ತಾ ಇದೀರಿ ಸರ್. ಚೇಂಜ್ ಮಾಡ್ಬೇಡ್ರಿ ಸರ್ ಪ್ಲೇಸ್. ಈಗ ದಯವಿಟ್ಟು ಅಡ್ಡಾಡ್ಬೇಡ್ರಿ. ಓಕೆ ಸರ್.
ಇಲ್ಲ ಅಡ್ಡಾಡ್ತಾ ಇಲ್ಲ, ಯಾಕೋ ನೆಟ್ವರ್ಕ್ ಇದಾಗ್ತಾಯಿತ್ತು. ಸರಿ ಬಿಡಿ.
ಓಕೆ
Shall we start then?
ಓಕೆ ಸರ್, ಗೋ ಅಹೆಡ್.
ಶಶಿಕಿರಣ್.
Yes, sir. Just a minute, sir.
Network. Shashikiran, shall I go ahead?
Yes, I'll just check with the YouTube stream, sir. Just a minute.
ಸರ್ ನಮಸ್ಕಾರ. ನಾನು ಮಂಜುನಾಥ್ ರೆಡ್ಡಿ ಸರ್. ಮಲ್ಲಿನಾಥ್ ಸರ್.
Manjunath sir, unmute your mic.
Namaste sir. Namaste. Welcome to webinar sir.
ಸರ್.
Manjunath sir, you can start. It's on live.
Okay. Good afternoon to all. Respected Dean sir, respected speakers of this particular national webinar, respected Professor and Head Department of Veterinary Microbiology, Veterinary College Bidar. I welcome you all to this particular national webinar on awareness to mitigate outbreaks of lumpy skin disease. So, with regard to this particular webinar, I would like to say a few things that Shashikiran, am I audible?
Yes, sir. Now you are audible, sir.
Yes, you are audible.
Okay, there is some connection issues. So, okay. So, good afternoon to all. First of all, I welcome you all wholeheartedly to this particular national webinar. As you all aware of the fact that lumpy skin disease is an infectious pox viral disease of cattle and buffaloes, which is caused by lumpy skin disease virus of the genus Capripox under the family Poxviridae. Though it is not a new disease across the globe, but it was an exotic disease to our country until November 2019. The first report of notification of LSD in the country came in from the state of Odisha on 18th November 2019. Then it was followed by Andhra Pradesh, Maharashtra, and so on. Now many states of the country are witnessing the disease outbreaks, especially during this particular rainy season, including our state, that is the state of Karnataka, especially from the districts of Bidar, Bellary, Shimoga, and other parts. So, which were reported during the month of October 2020, causing huge economic losses to the farmers and also dairy industry as a whole. So in this regard, Government of India on 19/11/2019 issued an advisory to all states of the country for control of LSD, as it is a disease of economic importance due to the losses caused by temporary milk reduction, temporary or permanent sterility in the bulls, damage to hides, and death, ultimately death due to secondary bacterial infections. Hence, this particular webinar has been organized to sensitize and update the knowledge regarding the disease by eminent expert scientists of National Institute among all the stakeholders who are actually involved in control of this disease outbreak. So, with that intention, this particular webinar has been organized. So, with regard to webinar, for kind information of all the participants, I would like to say a few things. Webinar comprises of 3 lectures, which will be delivered by 3 eminent experts of the particular disease. It will first will be delivered by Dr. SB Sudhakar, Senior Scientist, ICR NISHAD BHOPAL, on diagnostic approaches of LSD. Second one by Dr. Manjunath Reddy GB, Scientist from ICR NIVEDI, Bengaluru, on epidemiological aspects of LSD. That will be followed by the topic by name Therapeutic Approaches for LSD by Dr. Vivek or Kasaralikar, Professor and Head Department of VCC Veterinary College, Bidar. So ultimately, it will be followed by the question and answer session, which will be taken care by one of our technical person. So to start with, first of all, I would like to welcome our beloved Dean Sir, Dr. Dilip Kumar, to give welcome address. Over to you, sir. Dean sir.
Okay. Thank you, Dr. Manjunath. So I'm happy that on the 37th Foundation Day of Veterinary College, Bidar, we are having a webinar, especially on a very new emerging disease, you can say, and also a burning topic of the hour, we can say. So lumpy skin disease. When we were graduates, we have not studied much on this particular disease. So now it becomes important thing for us all of us that we have to know about this, because as it is a economic important disease and involving the livestock production and as well as livestock industry, you can say. So in this particular webinar, we have 3 eminent speakers with us. The first speaker, Dr. SB Sudhakar, Senior Scientist of ICAR NISHAD BHOPAL, Madhya Pradesh, who will be enlightening us on the diagnostic approaches for lumpy skin disease. Then we have another eminent scientist with us, Dr. Manjunath Reddy, Scientist of ICR NIVEDI, Bengaluru, of our own state. So he will be elaborating on epidemiological aspects of lumpy skin disease. Then one more senior professor faculty with us, our own college professor, Dr. Vivek R. Kasaralikar, Professor and Head of Department of Veterinary Clinical Complex, Bidar. So he will be enlightening us on the therapeutic approaches for the lumpy skin disease. So by these 3 lectures, I hope we'll be very clear about this disease and what to do and what not to do. We'll be able to understand. So I hope most of our graduates as well as from abroad also, they are attending this webinar. So wish you all the best and wish the webinar a grand success. Thank you. Thank you.
Thank you, sir. Thank you for your welcome address. Now, I request Dr. Bhagwantappa, Assistant Professor, Department of Veterinary Surgery, to introduce our first speaker, Shashi Bhushan Sudhakar, sir. Over to you, Dr. Bhagwantappa.
So is it audible, Sudhakar? Audible?
Yes, sir, you're audible, sir.
Yes. So very good afternoon to all the participants for the National Webinar on Lumpy Skin Disease. It's my pleasure to introduce the great speaker today, the brief biodata of the Dr. S.B. Sudhakar, Senior Scientist, NISHAD BHOPAL. Dr. Shashi Bhushan Sudhakar, presently working as Senior Scientist at ICAR National Institute of High Security Animal Disease, Bhopal, Madhya Pradesh. And sir, born on 1st March 1980 in Bihar, graduated from West Bengal University of Animal and Fisheries Sciences, Kolkata in 2002, and MEC from Bihar Veterinary College, Patna in 2004, and his PhD from IVRI in 2012. And later, sir joined services in 2009 and served as ICAR IVRI Mukteshwar up to 2014. Is noted as a scientist and academician in the field of veterinary sciences with specialization in veterinary microbiology, pathology, immunology, molecular epidemiology, diagnostics, vaccinology, and avian diseases. He has special interest in exotic and emerging animal diseases. He worked on Schmallenberg virus disease as an exotic ruminant disease. Prior to joining ICAR NISHAD, Dr. Sudhakar served as at IVRI Regional Station Mukteshwar, where he worked on the diagnosis and vaccine preparations of the PPR disease. He has contributed various research papers in national and international science journals of repute and authored 2 books and several manual and chapters for his credit. His scientific and research contributions have been duly recognized by several national awards, including ICAR Award, that is Rajendra Prasad Puraskar Award. He is the person who identified, isolated, and reported the emergence of lumpy skin disease in India for the first time in 2019 in India. So since then, he is working towards the development of diagnostic and development of vaccine for the lumpy skin disease. Hence, and today we have a right person with us to enlighten us about the lumpy skin disease, disease diagnostic aspect and other things. So once again, I thank you and I wish a grand success and fruitful outcome of the National Webinar on Lumpy Skin Disease. Thank you, one and all.
Thank you, Dr. Bhagwantappa, for briefing our eminent speaker, Dr. S.B. Sudhakar, sir. Now I welcome Dr. S.B. Sudhakar, sir, to go ahead with his talk. Welcome you, sir.
Thank you.
Sir, can you hear me, sir?
Yes, I can hear you. Yeah.
Yes, sir.
Okay, is it visible now, right?
Yes, sir. Yes.
Honorable Vice Chancellor, Professor S.D. Narayan Swami, Registrar Professor K.C. Birana, and Dean Professor Dilip Kumar, and the whole of the organizing committee, my fellow speakers, and all the participants joined to this webinar. I take pleasure in congratulating Veterinary College Bidar on its 37th Foundation Day and wish the college continued success in years to come. When I got contacted for this participation in this webinar, I suddenly recalled back a surprising event occurred in August 2019 in Odisha. A febrile nodular skin disease of cattle has been noticed by local veterinary officers in 5 districts like Mayurbhanj, Balasore, Kendrapara, Cuttack, and Bhadrak, and it was rapidly spreading. So, Government of India has at that time constituted a team of experts to investigate these outbreaks and and to identify the causative agent behind this outbreak. Then expert team including me has visited the relevant places in 5 districts of Orissa and seen the rampant spread of this unknown disease, nodular skin disease. At the time it was used to call, and we collected the samples and field data from the various local veterinarians and the farmers, and we brought the sample to ICR NISHAD BHOPAL. Here laboratory diagnosis was made based on the suggested clinical symptoms, and differential diagnosis were done as per the OI guidelines. Finally, based on the virus isolation, then nucleotide sequencing and phylogenetic analysis, it was concluded that the involvement of lumpy skin disease virus in those outbreaks. Since then, many states in India is being reported for the existence or outbreaks of lumpy skin disease, and still till date it is being reported from several districts of various states of India. Although capripox virus disease was considered endemic in India, like sheeppox virus disease and goatpox virus disease, but LSD in cattle was exotic. Now LSD is present in India, and it has got one better destination as India and causing severe economic losses. To stop the further spread of this disease, we need to have a target to restrict its spread from the epicenter and target its eradication from our country. To achieve this goal, we have to have better understanding of this virus and disease along with all its elements. So let us explore LSD along with emphasis on diagnostic approaches, which will provide you with the understanding of how to effectively deal with the lumpy skin diseases. Lumpy skin disease is a high-impact transboundary viral disease of cattle, and it has been categorized on the list of notifiable disease by World Organization of Animal Health due to its rapid transboundary spread capability and its substantial cattle production loss, which is caused by this virus, and due to this presence of the virus in a country which restricts the export of cattle and their products from those countries. This LSD disease is caused by LSD virus, which belongs to genus Capripox virus under the subfamily Cordopox viridae and family Poxviridae. This Capripox virus genus is known to cause most significant economical disease of ruminants, and this genus comprises of goatpox virus, sheeppox virus, and lumpy skin disease virus. LSD virus is a single linear molecule of double-stranded DNA of 151 kilobase genome, and approximately it is coding for 156 protein. This virus is antigenically and genetically very closely related to sheeppox virus and goatpox virus, and all the three virus is having 96% nucleotide sequence identity, but these three viruses are phylogenetically very distinct. Lumpy skin disease is very host specific and mainly affects cattle and rarely buffalo. Natural cases of buffalo has been reported from Egypt in 2020 from Nepal also. This disease is not reported from sheep and goat even when they are kept in close contact with the infected cattle populations. Clinical disease is reported in giraffe, impala, Arabian oryx, springbok, and as well as African antelopes. All breeds of cattle, both sexes and all ages, are susceptible to this disease. However, young ones may be more susceptible to the severe form of LSD. Bos taurus is more susceptible than Bos indicus, and Holstein Friesian and crossbred cattle having more prevalence of the disease than the Zebu cattle. Morbidity is reported to be between 5 to 45%. However, normally it occurs at 10%. However, but in certain cases it can go up to 85% also, which has been reported. Mortality is reported to be less than 10%. So in 2019, in Odisha outbreak, the morbidity was recorded as 7.1%, and so far, mortality from this disease in India is not been recorded. Existence of a specific reservoir or carrier status for this disease is not known. Incidences of disease can vary considerably depending on the presence of growing number of naive animals, immunological status of the host population, uncontrolled animal movement, abundance of vector population, virus strain. An outbreak is mostly seasonal, like in hot and humid climate or rainy season, the precipitation of disease will be more, but it has been seen that in many outbreaks or many outbreak regions, it can occur in any season because no season is completely vector free because it is vector driven disease. LSD is transmitted mainly by hematophagous arthropod vectors, and these vectors includes mosquitoes, ticks, flies, and probably midges, which has been variously been implicated in transmission of this disease. Various species of mosquito, and the potential prevalence of this vector population depends on, it varies from outbreak to outbreak because it depends on season and the vegetation which favors the breeding and development of these different species like Aedes aegypti, Culex, Rhipicephalus, Emblema, Hyaloma, and etc. It has been reported that the transmission can occur through these species also. Direct contact is mostly ineffective, but it can occur. However, indirect contact with the body fluids and skin lesion is reported to occur. And this virus is mostly excreted in semen, so it can transmit through artificial or natural insemination process. Intrauterine transfer from infected mother to calf can occur, and suckling calf may acquire infection from the infected mother also. Transmission may also occur iatrogenically during the treatment or mass vaccination in which single syringe or needle is being used for multiple animals. Long distance spread of this disease can occur due to the movement or transport of live infected animals from the infected area to free area due to trade, cultural festivities, seasonal grazing, nomadic farming practices. And Aedes aegypti has been implicated in long distance airborne spread of this disease also. Similarly, short distance spread can also occur due to the movement of animals, transport of animals, and within the 5 kilometer radius of the outbreak, this biting insect can transmit this disease aerially. Now, let us have a look on history and geographical distribution. This disease was originated in Africa, and from African subcontinent, it went to Middle East, and from Middle East, it went both sides, towards west to Europe and towards east to Asian countries and others. For a long time, a lumpy skin disease-like disease was affecting cattle in the Central African region for a long time. But it has been reported for the first time in 1929 from the Zambia particularly, in the southern African continent. And in the next 10 years, this by 1940, this virus has spread in almost all South African continents. And in the following decades, it has spread slowly towards northward, and it has and presently, the present state is all the African continents are affected by this disease. But surprisingly, Morocco, Algeria, Tunisia, and Libya are still free from this LSD. In 1988, from Egypt, first occurrence of LSD in Egypt was reported, and in the next year, in 1989, this disease went outside the African continent to Israel. And from 1990 onwards, this disease was roaming to all the Middle Eastern countries, and by now 2010 or 12, it has swept the whole of the Middle East. In late 2013 or initial 2014, the disease outbreak were reported from Turkey, Iran, Iraq, Azerbaijan, and part of Cyprus. And from Turkey, Later, in the later course of time, from Turkey swiftly spread towards the east to Balkans country through Greece and Bulgaria, and from there it spread towards east to Caucasus countries. Currently, LSD keeps on spreading to most South Asian countries. Like in 2015, first outbreak reported from Russian Federation, 2016 Kazakhstan, 2019 China, India, and Bangladesh simultaneously, and 2020 Nepal, Chinese Taipei, and very recently, last month, it has been reported from Bhutan for the first time. So it has originated from African continent, swept almost all the countries of African continent, and from there it went to Middle East. From Middle East, it went towards east to European countries and towards south to Asian countries. Now coming to clinical feature, the symptoms of LSD is mild to severe, characterized by high fever, around more than 40 degrees centigrade, and it may persist for 1 or 2 weeks. You can see the enlargement of subscapular and prefemoral lymph nodes. Edema of limbs and brisket are very common feature which can be easily recognized. Ocular and nasal discharges, which later becomes mucopurulent, and the appearance of skin lesions means in severe cases it can entire body may be covered with the skin nodules, but in mild cases the few skin nodules may be found in different parts of the body, but the exact predilection site you can't determine, because you may find all of the body or some of the body variably been affected with the observation of the skin nodules. These skin nodules are normally firm and slightly raised from the surrounding skins, which often separated by a narrow ring of hemorrhage. And in the later course, this narrow ring becomes necrotic, and the scab will sloughed off, leaving the full thickness of a hole in the skin. It may go up to the musculature layer. So characteristic inverted conical zone of necrosis, that is called sit-fast, which can be seen in most of the cases. Lesion may be observed throughout the digestive and respiratory tract. Corneal opacity or blindness may be in some cases unilateral or bilateral keratitis, and in male animals, orchitis is often seen. This disease can be a silent or subclinical type also. In affected cattle exhibit lameness, emaciation, milk production loss, and pneumonia if respiratory tract is being involved. Lumpy skin disease results in overwhelming economic losses, which impose dramatic effect on rural livelihood, which is strongly supported by cattle production. Direct losses are due to emaciation, damaged carcass, decrease in milk production, and decrease in meat production also, mastitis, fertility problems and abortions, veterinary treatment cost and deaths, damaged skin and hides. Disease consequences are very devastating at national level. The cost incurred may include some or all of the following: awareness campaigns, monitoring and surveillance programs, sampling and laboratory testing, vector control, culling or carcass disposal, compensation program for culled or slaughtered animal. This depends on the policy of the government. Cleansing and disinfection of affected farms, purchase of vaccines and implementation of a vaccination program, a restriction on movement of live animals in its product, and ban on export of live animals and its products. According to a report by Gary et al. In 2011 from Ethiopia, financial loss estimated per head was for local Jebu cattle $6.43 US, and for Holstein Friesian and Crossway it was $58 US. If you convert this figure for at the present time, it may come around 500 for Jebu cattle and around less than 5,000 for Holstein Friesian and Crossbred cattle. Now coming to diagnosis. Diagnosis of LSD can be established based on characteristic clinical sign and laboratory confirmation for the presence of virus or viral antigens. A detailed history of the disease must be collected from different sources, and these are the source of information which can help to collect the data from the field levels, like interaction with the farmers and other workings with the livestock on the premises, record of cattle movement kept by farmers, then veterinarians record treatment and vaccination, record of cattle mortality and sick animal, and laboratory test results, if any, cattle traders record, and reports from slaughterhouse is also important, because most of the cases post-mortem is not being done for this in this outbreak if mortality also happened. A number of laboratory tests are available for diagnosis of lumpy skin disease, virus, for detection of viral antigen, and for antibodies, such as virus isolation, molecular methods like all PCRs, virus neutralization test, ELISA, fluorescent antibody test, EGID, IPMA, etc. Laboratory confirmation of LSD employing different methods requires appropriate collection of samples like tissue, blood, semen, serum, etc. Now, which type of sample and how the sample should be collected? So, let us have some brief or detail on the sample collection, sample types, and sample segment procedures. During sample collection, sample general guidelines should be followed, like proper authority must be informed about the disease. Protective clothing must be used to ensure good biosecurity practices. Work aseptically to avoid cross-contamination between the two samples. Disinfect the sample collection site. Change needle, scalpel, and gloves between the two animals. Avoid undue stress or injury to animals during sampling. That's why proper sedating or restraining is required to collect the samples. If many number of animals are present in a herd and few animals are affected, so separate the affected or suspected case and then collect the samples from the rest of the herd. Collect at least 5 samples if multiple animals are affected, but however, more number of animals are sampled, the better, in order to detect those animals that are incubating the disease. During sampling, better consider duplicate number of samples. One set can be sent to referral diagnostic laboratory, and one set can be preserved for future use. And samples should be labeled with the waterproof marker pen, and organized to transport this collected sample without delay. And maintenance of cold chain is always advisable. And before dispatching the sample, you should inform the laboratory about the sample sending. Several commercial viral transport medias are available in the market. Alternatively, a sterile phosphate buffer saline with antibiotics can be used if commercial BTM is not available at the point. That sample types, preferred sample types, it is very important to collect the sample at appropriate stage of disease. Like for skin nodular lesion or scabs, collect a skin biopsy from skin nodules, preferably within the first or second week of appearance of clinical signs. These samples are suitable for detection of viral DNA or virus isolation. Whole thickness of a skin sample should be collected using local anesthesia or appropriate surgical techniques. Whole blood sample can be collected during the febrile stage of the disease, which is normally from 2 to 7 days of appearance of clinical sign. In EDTA tube, you can collect blood for PCR detection, or if virus isolation is the target, in heparin, Vicodin can be used. Paired sera sample at acute and convalescent stage of disease can be collected for detection of LSDV antibody by serum or virus neutralization test or IPMA. Ocular, nasal, or oral swabs can be collected for antigen detection when these animals are showing such discharges, which normally animal will show this discharges from 2 to 3 weeks of first appearance of clinical signs. Apart from these samples, if breeding bulls are involved in the outbreak, so you can collect 2-3 ml of raw semen, preferably within 2 to 3 weeks of appearance of clinical sign, because this virus is reported to be excreted in semen also, and it can transmit through insemination, either natural or artificial. So semen testing is also very important. For sample shipment, national guidelines must be followed while transporting the sample to laboratory, even if it is carried by a special messenger. Inform the laboratory before sending the samples, and samples should be labeled properly with following information: sample ID number, dates, species, sample type, or location of the outbreak, and any other information relevant to this outbreak. The sample must be accompanied by a sample submission form. Always a sample submission form is available for individual disease. So all the columns or all the information which are queried in the sample submission form should be duly filled, which will help on achieving the confirmatory diagnosis. Like maintenance of cold chain is essential for sample quality. Store the sample at refrigerated temperature 4 degrees C until shipping, and shipping in ice and gel pack is recommended for 24-hour transport. If shipping period is more than 48 hours, you better ship it in dry ice to maintain the quality of samples. For packaging of these samples, triple layer leak-proof packing structure is required, in which sample infectious substance or sample containers should be enclosed in primary or secondary robust leak-proof watertight containers, receptacles with absorbent packing material in between them, and after that tertiary rigid outer packaging is being done, so that in transport the sample container will not be damaged, or there will no loss of sample, no leakage, and all the things.
Excuse me, sir.
Yeah, please.
So can you please share the screen once again, sir, because yeah, some people are not able to see it. Can you just stop the presentation and just start once again, sir?
How to stop? I stop screen sharing, right?
Yes, sir. Yes, sir. You stop.
Yeah, I stopped. So I will present now again.
Yes, sir. You just once again, you just present.
Entire screen.
Last 2 slides, sir.
Share. Is it coming now?
Yes, sir.
Yes, sir. Yes, sir. We can see, sir.
Okay. So I was talking about sample shipment. For shipment of sample, I will repeat this slide again. National guidelines must be followed while shipping the samples, even if it is carried by a special messenger. Inform the laboratory before sending the sample, so that laboratory will have proper time to immediately process and test the samples. Samples should be labeled properly before they dispatch with the following information: sample ID number. It will be neatly written: date, species, sample type, and location of the collection, and any other relevant information. Sample must be accompanied by sample submission form, which may be kept one copy inside the carton and another copy over the carton. Maintenance of cold chain is essential for sample quality. Store the sample at refrigerated temperature until shipping in ice and gel pack if shipping time is less than 24 hours. But if shipping time exceeds 24 hours or more than 48 hours, better to ship in dry ice, so that sample quality will not be deteriorated. For packaging, normally we use triple layer leak-proof packing structure. Like sample will be enclosed in primary and secondary robust leak-proof and watertight receptacles, and with absorbent material in between them, and then tertiary rigid outer packing is being done. So severe cases of lumpy skin disease are highly characteristic and it can be easily recognized, but mild cases and subclinical infection may confuse with the other diseases of skin. The following diseases may be considered for differential diagnosis of LSD. First one is pseudolumpy skin disease. It is caused by bovine herpes virus 2. It has two forms. One pseudolumpy skin disease and another form is bovine mammallitis. Bovine mammallitis mostly lesion restricted to udder and teats. And pseudolumpy skin, that superficial lesion of shorter duration and less severe form, can occur at neck, shoulder and other parts, but it is of shorter duration. Bovine papillary stomatitis, it is a parapox virus. and lesion mostly occur in oral mucous membrane, lips and muzzle. It mostly affects the bovine population of less than 2 years of age, and it is mostly mild in nature.
So sorry to interrupt. We are unable to hear your voice, sir.
Hello?
Sudhakar, sir.
Yes. Are you able to hear me? You're presenting to stop presenting. Hello?
Sir.
Yes, sir.
Again, you
Yes,
please
sir.
share
Yes.
the.
Is it audible?
It is audible,
Audible.
sir. Please share. I think you'll remove the slide sharing, sir.
Yeah, I have removed that one.
Yes.
Okay.
Please share again, sir.
Yeah. It is coming now?
Yes, sir. Yes, sir.
Okay. What is happening?
Sir, make it slideshow, sir. Make it slideshow.
Yeah. Okay.
Now it is.
So we were discussing about the differential diagnosis of the lumpy skin disease. So pseudococcopox, it is also a parapox virus, but lesion mainly occur on udder and kids and develop as a blisters. Insect bite, urticarial, and photosensitization. Lesions will be more superficial, shorter duration, and less severe. Early ringworm lesion or dermatophillosis, these are bacterial or parasitic origin. Red, itchy, scaly, circular rashes will be seen on the body surface. Hair loss will also occur and will be more superficial, and dry, non-ulcerative surfaces will be seen in these cases. Dermaticosis is caused by Dermotex mites. Papules and nodules over the withers, neck, back, and flanks will be observed with alopecia, and you can suspect mite presence on the lesion also. Onchocercosis, it is due to nematode parasites. A lesion like small nodules, mostly at ventral midline and udder. Besnoichiosis, it is a protozoan disease in which we can find thickened and folded skin with alopecia, scleroderma, and hyperkeratosis. Cyst may be found on conjunctiva or genital organs. And vaccine reaction in live attenuated vaccine, homologous vaccine, there will be some swelling at the inoculation site, and in another form, small nodules you can find in general body surface, which is known as Nithling disease. However, this may regress within a week. And all these different diseases can be ruled out by detecting LSDB by PCR in our samples. Now here comes the OIE recommended test. Several tests has been recommended by OIE for different purposes. But in India, we are mostly related to the confirmation of clinical case. So virus isolation and PCR is the recommended method. And for antibody detection, virus neutralization test is a recommended method. However, during the later course of time when India will be trying for eradication of disease, so for those purposes, OIE has recommended the exact tests. Virus isolation is the method to investigate the viability of virus in the samples for its identification and characterization. This LSD virus grows on tissue culture of bovine, or caprine origin. Primary and secondary cell lines are most sensitive. Primary and secondary cells are most sensitive cell cultures. However, the continuous cell lines also can be used. Primary cells like lamb testicles or lamb kidney cells, continuous cell lines like MDBK, Vero, OA3.TS cells can be used for isolation of virus. This virus can also be isolated in embryonated chicken egg of 7 to 11 day old through cam route. In cell culture, the virus manifests itself by cytopathic effect, and in the embryonated chicken egg, development of pock lesion on the chorioallantoic membrane. The virus, mostly pox virus, grows very slowly, which requires several passages for a characteristic CP to observe. That's why this PCR assay has replaced the virus isolation for primary diagnostic test. Electron microscopy can also be used to identify lumpy skin disease virus, but differentiation between the capripox viruses and other orthopox viruses is not possible. And this picture shows the cytopathic changes produced by LSDB isolated in primary LT cell culture. This sample was from Orissa, and in LT cell it was isolated. So aggregation and clumping of cells and detachment of cells will be like a cytopathic effect. Now coming to the molecular methods, viral genome detection is an indirect proof of virus presence, which can be achieved by PCR-based technology. Several highly sensitive, well-validated PCR methods are developed using different targets, different multiplication systems, and different visualization methods like in-point or real-time or naked eye visualization. These tests have also been developed with different aims, such as detecting all capripox viruses at once, like SIPpox, GOTpox, and LSD at once, and capripox genus-specific detection, differentiation of all the three capripox viruses, differentiation of wild type versus vaccine virus, LSD virus, and multi-disease detection or the combination of all these above. Gel-based PCR are widely used by basic method for LSD diagnosis. It is also very recommended. This is reliable, sensitive, and very good backup method in every lab. However, real-time PCR is faster, sensitive, and less prone to contamination. Both the tests can be equally useful. Now coming to pan-capripox PCR means at a single test you can detect the capripox, SIPpox, GOTpox, and lumpy skin disease virus in one test. Because genetic similarity of these three members of capripox genus has enabled to develop rapid, sensitive, and well-validated conventional and real-time group-specific PCR. Several gel-based pan-capripox PCR was developed targeting various genes, like virus attachment gene that was reported by Ireland and BHOPAL, that has been also recommended by OIE for LSD diagnosis, and another thymidine kinase gene were used for development of a gel-based PCR by Tegalin in 2014. Any gene can be targeted and validated, then it can be used for the diagnosis. In real time, likewise, several TACMAN-based real-time capripox PCR was developed targeting different genes like polymerase gene by Balansky, then ORF74, that is attachment gene by Borden et al. And these two real-time PCR has also been recommended by OI for detection of LSDB and the capripox viruses. In 2013, again, Eggman has developed the real-time PCR targeting three different regions in a viral genome like D5R, E3L, J6R. In most of the laboratory LSD diagnoses are based on the generalized detection of capripox virus, but it cannot differentiate between LSD, SIPpox, and GOTpox virus. A species-specific assay are developed for this separately for SIPpox, GOTpox, and LSD, which are of value when the clinical sign of LSD is being reported or observed in the cattle vaccinated with SIPpox virus vaccine or GOTpox virus vaccine, or in other case, if all the three capripox virus diseases are present in the country, like the present situation in India. So several gel-based PCR which can differentiate three members of capripox genus, combining it with RFLP or targeting one species specifically. Bean et al. Has developed this PCR RFLP assay targeting P32 gene, where different restriction enzyme has been used to differentiate SIPpox, GOTpox, and lumpy skin disease virus. Different patterns were seen. Another approach is to use a feature. In RPO30 gene of capripox virus, there is a 21 base pair deletion. So the different virus. So here there is no need of additional digestion of with restriction engine. It has been developed in 2011 by UNIA. LSD specific assay has also been developed, targeting genome termini and ORF 132. Similarly, real-time PCR was also developed for differentiation of these three members of Capripox viruses. Close-sense resonance energy transfer principle for construction of probes. A real-time assay is developed targeting GPCR gene for differentiation. There is difference in the GPCR gene of all the three species of this virus, and the melting point temperature difference of the amplicon is being monitored by melting curve analysis, which will give the three different peaks of this virus, of these three viruses. Similarly, using this melting curve analysis, an assay was developed to differentiate 8 pox viruses belongs to 3 genera: orthopox, capripox, and parapox. Orthopox included that's cowpox and camelpox. Capripox like SPBV, SIPpox, goatpox, and LSDB, and parapox like pseudocowpox, bovine papillary stomatitis. It has been developed recently in 2017 by Gile et al. Now coming to DIVA strategy for detection of field virus and vaccine strain virus. So when a characteristic clinical sign or symptoms of LSD is being observed in the cattle vaccinated with vaccine containing live attenuated LSDV, several molecular tests have been developed to identify the etiological agent, whether the field virus has a role in this disease or vaccine virus itself is causing the adverse reaction. So for this also, nested and gradient PCR RFLP was developed to classify wild type and vaccine LSDV virus. Most of the real-time PCR developed for DIVA strategy exploited the feature that the vaccine strain of this LSDB has 27 base pair deletion in EEB gene, LSDB 126 gene. So this strategy has been used for development of HRM curve assay. Pacman based 2 real-time PCR was also specifically used for detection of virulent LSDB strain in mixed wild type or vaccine type by Beganovic. Additionally, a duplex Pacman assay targeting GPCR gene for both wild type and LSD vaccine strain was developed, and one specifically for methylene based vaccine targeting LSDB 008 gene. And beyond this, an HRM curve assay for detection and differentiation among SIPpox, goatpox, and LSDB, the two in LSDB field strain and vaccine strain, was developed and validated by Pestova et al. Recently in 2018. Now some other test was also available for detection of LSD. Loop mediated isothermal amplification assay. It is a derivative of classical PCR. This isothermal assay has advantage from equipment point of view, and result can also be visualized by seeing the turbidity, colorimetry, or agarol gel also can be used. Based on this technology, a pan-Capripox lamp was developed by Das et al. In 2012, and they were using a polymerase gene, and based on P32 gene, that is attachment gene, Murray has developed and evaluated another lamp assay. Similarly, recombinase polymerase amplification assay is another isothermal process which has been used to detect Capripox viruses rapidly. Recently, LSDB RPA-based method using FAM-modified probes for real-time detection reported with analytical sensitivity of around 180 copies and 100% diagnostic specificity and sensitivity. LAMP assay has another great advantage that it can be developed in a lateral flow assay like Pennside kit, which can be administered easily at field level. Now coming to serology, in general, the immune a status of a previously infected or vaccinated cattle cannot directly be related to the serum levels of antibody, because in LSDB infection, cell-mediated immune response is predominant than the humoral response. However, a number of methods have been employed for serological investigation of lumpy skin disease virus. Serum or virus neutralization test is a gold standard recommended by OI. Alternatively, AGID and fluorescent antibody test can also be used, but AGID is less sensitive and cross-react with other parapox viruses also. Fluorescent antibody test needs meaningful optimization and validation. Western blots are sensitive, specific, but difficult and expensive to carry out at each laboratory. Immunoperoxidase monolayer assay recently developed by Hegman et al. In 2020. It is relatively easy to use and sensitive and specific, but it involves the working with the cell culture and live viruses. ELISA technique is very laboratory friendly, and recently a commercial ELISA kit. However, it can detect all the 3 capripox viruses. It has used double antigen technology for detection of multi-species sample. It is available from ID, but with better sensitivity and specificity. But at the financial side, it may be expensive for developing countries. Molecular characterization can be achieved by sequencing and phylogenetic studies with targeting complete genome, individual genes, partial genome fragments, with the aim to trace the origin of virus, to study the evolution of the virus, and to differentiate 3 capripox viruses, LSD vaccine strain and field strain. And this is the phylogenetic tree of the Indian Orissa outbreak. These 5 isolate were put for nucleotide sequencing initially with the attachment gene, partial attachment gene, and it has been clustered. It is all the 5 isolate were clustered with the field strain of LSD. To do the characterization molecular study sequencing of phylogenetic study, data analysis required extensive learning. That's why sequencing is not the technology suitable for routine screening in each lab. But it gives the major information about the origin evolution of this virus. Now, several commercial real-time PCR kits are available in the market for detection of LSDB. Some kits are also able to differentiate all the 3 capripox viruses, and some LSD DIVA kit is also available in the market, which is beneficial for diagnosis purpose. Like Technique, Genesis, Tetracore, InGenetic, these brands are launched their kits in the market. LSD has no treatment, no specific treatment. Only symptomatic treatment can be tried using antimicrobials, anti-inflammatory, antiseptic solutions, and good nursing care. Animal infected with LSD virus generally recover. Moreover, the treatment and complication of LSD is very costly. That's why prevention is more beneficial in order to curtail the economic losses. So, recommended actions are: suspected animals should be isolated from rest of the herd, and the farm should be quarantined till definitive diagnosis is determined. During outbreak or in a high-risk area, cattle should be monitored daily for development of any characteristic clinical signs. When LSD is suspected, cattle movement should be stopped establish a nationwide laboratory network for laboratory diagnosis. There is a need to develop a pen-side kit where it can be easily administered at field level. Development of homologous vaccine against LSD is very much required. However, we are using the heterologous vaccine that also protects cattle against LSD, but it gives only partial protection by several reports. There is a strong commitment for effective implementation of prevention and control measures. As I said, each month in India, a new state is being reported for presence of LSD. So there definitely needs a strong commitment from all the stakeholders for effective implementation of this strategy. Here is some link for advisory on lumpy skin disease, referral laboratory for LSD in India, guidelines for sample collection and dispatch for diagnosis of lumpy skin disease, format of sample submission form for lumpy skin disease. Thanks. Yeah, shall I stop presenting?
Yes, sir. Yes, sir. Sir, thank you, sir. Thank you very much for wonderful talk, sir. I think you have covered many things and you have not left anything for other speakers to speak.
Yeah, yes.
Yes, sir.
Give some more information also.
Yes, sir. Yes, sir. It is required, sir. And we wholeheartedly thank you for giving wonderful talk, sir. And I request you to be online, sir. We will be taking many questions have come in. We will be taking at the end, sir. So other speakers are waiting for their this thing, sir. So thank you, sir. And now I request Dr. Arun Karate to introduce our second speaker, Dr. Manjunath Reddy, who is speaking on of LSD. So over to you, Dr. Arun Karate.
Thank you, sir. Good afternoon to all. It gives me immense pleasure to introduce Dr. G.B. Manjunath Reddy. After his BVSc and AH at Bengaluru, Dr. G.B. Manjunath Reddy completed his MUSc and PhD in veterinary pathology. Presently, he is working as a scientist at ICAR, NIVEDI. His area of research is epidemiology and pathology of pox viral diseases of animals and rabies. About his research projects, he has completed 6 externally funded projects and 8 institutional funded projects. He is now undertaking 8 ongoing projects, of which 4 are externally funded projects and 4 are institutional funded. Coming to awards, he has received many awards, out of which few I will mention here. He has won Young Scientist Award, Best Doctoral Thesis Award, 3 Best Research Article Award, 5 Best Poster Award, and 5 Best Oral Presentation. He has also won DST International Travel Grant Award, Denmark, and NAIP International Travel Grant Award, France. About students guided, he has guided 2 National Postdoctoral Fellows, and he has completed as a guide 8 master students he has guided, and for 8 students he was member, and he has guided 2 doctoral students, and for 3 doctoral students he was member. And coming to publications, he has published 64 research articles, 7 review articles, 32 invited papers, 10 popular articles, 297 gene bank accessions, 1 whole genome, 64 research abstract. He has also written 1 book, and he has written 11 chapters in books, and he has published 20 technical bulletins, 6 training manuals, and 23 training he has organized. This was in brief about Dr. G.B. Manjunath Reddy. We are very happy to have you here, sir. Thank you, one and all. Over to Dr. Manjunath, sir, please.
Arun Karate for briefing biodata of Dr. Manjunath Reddy, GB, sir. Now I request Dr. Manjunath Reddy, sir, to present his talk that is on epidemiological aspects of lumpy skin disease. Over to you, sir. Reddy, sir.
Yes. Good afternoon to all of you. First, I would like to thank BC sir and the Dean Bidar Veterinary College and Manjunath sir as well as my colleague Arun for giving me an opportunity to participate as well as to present about the epidemiological aspects of LSTV. Can you see my this one, sir? PPT.
Yes, sir. It's visible, sir.
It's visible, right?
Yes, sir.
Okay. So, myself, Dr. Manjunath Reddy. I am working as a scientist at ICR NIVEDI. Before in NIVEDI, I was at CIRG in Makdoum and I worked on goat health.
Sir, it is not getting uploaded, sir.
It's not getting uploaded? Sir?
Oh, it's visible, sir. We can see your presentation, sir.
It is visible, right? Sir, Malina sir is telling you cannot see. Malina sir, is it okay? Hello? Is it okay?
Yes, sir. Yes,
Network, sir. My network.
it is visible.
Sir, most probably.
It is visible, sir. You go ahead.
Okay. Yeah.
No, I'm unable to see, sir. I'm unable to see, sir.
ನಿಮ್ದು most probably network ಸ್ವಲ್ಪ ಸ್ಲೋ ಇರಬೇಕು ಅನ್ಸುತ್ತೆ ಸರ್.
Yes, it's good. It can be seen.
So before, shall I continue, sir?
You can continue, please.
Yeah. Okay. Before I directly jump into the LSDV, just I would like to say you that whatever we do as a clinicians or basic veterinarians, we come across many times symptoms. Certain symptoms, they become syndromes. Once today's syndrome, it is going to be a tomorrow's disease. That's how LSDV also started. If you look at the papers and all those things, they started with the naming as a nodular meningitis, even in Odisha itself. Then they called it as a dermatitis. Then it became a capripox. Instead of telling it is LSDV, they said it is in bovine. Then they narrowed down to capripox virus infection cattle. Then finally they said it is LSDV. This is a brief. Why it created so much havoc, or it's kind of an emergency, like the coronavirus in humans. This has come at a similar time where that diseases or in general viruses, diseases, the general public is aware. Now even go to Karnataka or anywhere, you ask this disease, they'll compare with corona of animals, even though it is a pox virus. That is a layman's language. If you look at the papers, because of this much publicity, there is a pressure or our responsibility as a veterinarians, whether you are a doctor or you are at the diagnostic level or you are at the policy level or you are at a teaching level, that made us to much more responsible to look into the animal health in terms of diseases. So I'll go to the LSD, just an introduction where already Sudhakar sir has said. He made my presentation a bit easy when compared to, otherwise I would have felt a bit difficulty. So the introductory part, as many of the students are also participating, the lumpy skin disease is a pox virus disease. It affects the cattle and buffalo. Maximum it affects the cattle. Because I underwent a lot of outbreak investigation, I could not find any buffalo got affected. There is something which might be making this virus, which is loving or it is attracting towards the cattle. Why it is so much important? Because the zebu cattle, or we can call it native breeds, it is affecting more native breeds. The moment it affects the native breeds are held by the poor farmers who keeps them as a livelihood security. Or in Karnataka, if you go to the North Karnataka portion, they keep the bullocks for the purpose of drought purpose. The moment this disease affects the drought power, he cannot no more plow the field, or he cannot do the agriculture, where it is a bread and butter. That's how this disease has gained so much importance, not only because of milk production, it is because it is affecting drought power of the other animals, including the milk production. However, if you compare the mastitis, all these things, it is an endemic or it is going on. It has captured so much importance because it affecting both the drought power as well as the production in terms of milk or in terms of hide quality, or sometimes it causes even abortions because we also came across one of the case wherein the abortion, even the fetus showed some of the pock lesions. Some infertility is also reported. The direct losses. The indirect losses includes the restriction in the movement of the animal itself, in terms of whether you are going to sell it or the entry into the other market area or the purchase by the other farmer. If your bullock or your cattle got infected, the value or the price you are going to fetch, it automatically reduces, or even sometimes they reduce by 50%. That's called as a distress sale. So along with that, there is a restriction on the trade, which includes whether it is a hide or even meat. All these things, whether it is national trade or interstate or international trade. That's how the disease has gained more importance. And when it comes to the historically, as already Sudhakar sir said, it was restricted to African countries. Maximum it was in the Eastern and South Africa. Why this disease jumped from there to here? See, first it was in native breeds. Gradually, if you look at it came towards the European countries, then it jumped into the dairy industry. Before that, in the African countries, there are local breeds. It could not gain that much importance because it did not affect any milk production. So there the people used to keep the animals for the livelihood as a meat or sometimes hide, all these things. When it comes to Europe and the countries like India, where the animals are mainly kept for milk production, the moment the milk production is reduced, you are under the hot seat to control the disease. That's how this disease has gradually inculcated or entered the other parts of the country, other parts of the continents as well as the world. Maybe it is going to enter some more other countries. For that, OIE or even WHO, everybody is behind us to control the disease. So typically, LSDV outbreaks, they does not occur very frequently, like your FMDV or some of the very less like enterotoxemia, all these things, because it is a very adamant virus which has a good immunity power, and it occurs at several year gaps apart. But there are many epidemiological gaps why it occurs with a duration of 5 years or 7 years, or how the virus survives in the environment before it causes the outbreaks. That it is still has to be understood. And usually a primary case, see for example in Karnataka, the primary case, it occurred or it might have occurred in certain place. It is maximum or many times it is due to an import or illegal movement of the animal. It's rarely by the transportation or long distance. It's the host which moves along with the vectors. Then, as already Sudhakar said, it is a notifiable disease. That means once the disease is there, you are bound to notify it to OIE. Once you are notified, there are certain restrictions on your country trade also. So the disease outbreaks in India, first time as Sudhakar and his team, they went and investigated 2019. But we utterly failed to control the disease. From there, it is now spread all over the country almost. Now it has become an animal emergency. like in 2020, if you look at the population of the cattle and buffalo, the right side of the maps, there is a huge population and susceptible. So how to protect them? That is our job. So until unless you don't understand the disease, you can't protect the disease. So I will deal in maybe 3 terms of the epidemiology of this disease. Host followed by pathogen, or how environment plays their role. Because we work in Epidemiology Institute, that's how we deal the any kind of disease. First, I'll go to pathogen. Sir has explained, it comes under the pox viridae, all these things. But this disease, the pox virus, is one of the biggest virus, but the host range is very less when compared to other viruses. If you take the rabies example, even though it is the genotype 1, it has long host range, whereas the pox viruses are very, very restrictive in nature. Even if you take out the sheep pox or goat pox and LSDB, why this happens? Because of the structure of the genome. If you look at the genome, it's the terminal portions will have this host range genes, because the location of these genes makes this virus more host specific rather than a broad host specific, and it gives us more opportunity to pick the virus easily when you go for any kind of identification tests. And also there is less need of development of specific diagnosis or a specific assays. That is an advantage of having the poxviral diseases also. And as we are already told, this virus grows very slow, very, very slow, because we are growing in our own laboratory many times this sheep and goat pox, and it requires either bovine or bovine, usually primary cell cultures. However, we have cell lines which can grow the virus. This is of research importance, maybe not much of helpful for the field veterinarians. And for the students, it is a DNA virus, obviously. It causes the endocytoplasmic inclusions. When you examine an histopathology slide or a cell culture, if you stain it, it's a beautiful inclusions you can find, which gives a clue for diagnosis. And when it comes to the virulence, there is no clear-cut difference in the virulence between strains, because there is only one strain which exists, either field strain or it is a vaccine strain. In case of LSDB, in case of sheep and goat pox also, it's there are less number of strains. Unlike your blue tongue virus or some other like FMDV or foot and mouth disease virus, where you have more strains, hence the virulence is varied. Here there is no much virulence variation, and between the host also there is very less, or breed wise or stage wise. However, the virus is a bit susceptible to temperature when compared to other viruses, where they don't have envelope, because this virus is having envelope, a good envelope. Because of that, as well as the scab formation, which creates a fort, or it is a covering under which the virus can survive for a long time. Even if it is exposed to the sunlight, it can survive for a longer duration. And if you preserve it in minus 80, or for a research purpose, you can store the virus as such with a scab maybe up to 10 years, and at 4 degrees even 6 months. And pH also, it is not that much susceptible at the neutral range, whereas too much alkali or too much acidic, it is susceptible because of its envelope nature. And obviously there are certain disinfectants which chloroform, all these are lipid solvents, because as I said, it is an enveloped virus. You can use lipid solvents as a inactivation agents. These are properties some of the viruses. Because these viruses, as already said, they have been classified into capripox, the LSDV. It is only one serotype, because they are all sheep pox, then goat pox, then lumpy skin disease. They are all serologically cross-reactive. Unlike the other diseases, like example is foot and mouth disease, where you can trace from one the second strategy to go for only good bugs at present is to avoid intermixing of vaccine strain with the field strain, because these are all attenuated viruses. They do sometimes cause a little bit of mild clinical signs, including sometimes you find epoch lesions, mild epoch lesions and fever, all these things. That's how they advise the goatpox vaccine. Depending on the phylogenetic, this one relatedness, they advise the goatpox in India. That was with one gene. There is one more gene called RPO30. This also classifies these viruses beautifully into separate groups. These are our studies. Again, we did some LSDV sequencing also. It is closely matched with the goatpoxes. As of 2019, if you look at the map of the world, maximum of these countries like American, those are free, and European and Asia, it's quite a huge problem. This is again the recent one, 2020. The outbreaks are how the disease distributed spatially on different continents. This is proportion of outbreaks. How many outbreaks have occurred in 2019 in different countries. India is obviously contributing quite a lot. In 2020, if you look at this slide, I just put it, there are so many states. Blue color indicates that these are the states which have reported the disease with the help of diagnosis from NISHAD. And some of them, after the diagnosis from NISHAD, they do contact NIVEDI. And the right side is Karnataka. Many districts in Karnataka, the LSDVs outbreaks are ongoing also. Some have subsided. Some districts are reporting for the first time also. Then comes the transmission. Until unless you don't understand the transmission, control is difficult. And what I mean to say is, if you cut the transmission cycle, that's our job is done. So how this virus is getting transmitted? Mainly it is a mechanical transmission by vectors. What I mean to say mechanical vectors is, unlike blue tongue virus, where the virus will replicate in the vector, here the vectors like our flies, whether it is a stable fly, horn fly, or mosquitoes, Aedes, or ticks, in that the virus is not getting multiplied. It will be there at the maximum 2-3 days, and because of intermittent biting, they have a behavior of biting here one animal, then sucking the blood and going some other animal. That's how the disease is getting transmitted. Then there are other routes like saliva or nasal discharges. If you look at the picture, where the lesions are on the muzzle as well as almost inside the nostril, this makes the animal to secrete more nasal discharge as well as inside the gums. Once it goes grazing or it uses the same common sharing powder or water, these things, that makes the other animals also getting a source of infection. Then if the bull, or whether it is a breeding bulls or semen production bulls, if they are infected, they have shown that it is transmitted through either natural service or through artificial insemination. Then there is a high probability of vertical transmission. As I told you, some abortions cases what we observed after vaccination, they could see the pox lesions on the placenta of young one. Then viremia stays for only a few days, but in severe cases it may go up to 2 weeks. That makes the control very difficult, because the moment you have more severe case in an epidemiological unit like a village, it acts as a source for 2 weeks, controlling the vectors. It is quite a hectic job for a veterinarian or the owner itself. Then an infected pregnant, obviously, as I said, it will abort, and the lesions are beautiful on the surface of placenta. The virus, sometimes it is been proved that the suckling cows, they get infected through milk or from the skin lesions on the teats. I'll show you some of the next photos where you can see the skin lesions on the teats also. Then when it comes to the first, as I said, many of the outbreaks were traced to the first outbreak, what we call it as an epidemiological foci or epicenter, was traced back to some of the illegal movement or recent movement of the animal inside that village, or even sometimes the incubation period is up to 5 weeks. If you bring an animal after 1 month also, sometimes they do develop the diseases, or the movement of trucks or all these things. Then the long distances, as I said in the previous transmission cycle, long distance is attributed to cattle, either illegal transmission of the cattle or transportation. If you look at the mid one picture, where the bullock, it has been used, we took this photograph in Mandya, sugar factories, where they take that sugar cane for the factory delivery. The farmer used the affected bullock for the transportation of the sugarcane. He may not be aware of that, but if you look at that in and around the sugarcane factory, there were more than 100 bullock carts. That means 200 bullocks. The one village, one bullock is coming, 100 villages. Imagine the spread of the virus. So that's what I mean to say. A long distance travel by either cattle or vehicles. The short distance travels are usually with the vectors. This stomach says you see the blood-filled abdomen. Then as the behavior of the vector is frequently, the moment it takes the blood from one animal, the vectors have that affinity. Like we do like to taste different items in a plate of dinner or lunch. Definitely they'll have that habit of feeding on different animals in the same herd. So the changing host frequently, that makes the spread of the disease more faster. And as I said, it is not a biological vector, and the principal vectors definitely varies with the geography. See, towards, if you go to the coastal area, the vector may be different. If you come to the dry areas like Kolar or Chikbadapur, the vector may be different because it depends on the availability of the breeding grounds for the vectors, as well as suitable conditions for the vectors. Then when it comes to susceptible host, because host plays a very important role, as already my previous speaker said, cattle is the susceptible host. However, there are some wild animals which are also susceptible. Buffalo is also susceptible. At present, or as per my knowledge, buffalo, we could not get any case. The morbidity, obviously, it is quite huge, or quite high, up to even it go to 45%. Mortality is very rare. The mortality due to some other secondary bacterial infections or complications caused by LSDV or maggot wounds or this cataract, all these things are secondary bacterial infections cause mortality, especially in young ones, are where the immunosuppression or the high producing or high yielding cows, they're already under the stress of milk production. once the LSDV got infected, again the secondary bacterial infection, that complications only, very few deaths are reported. They have reported that it may replicate in sheep and goat. However, sheep and goat are not reservoirs for the LSDV, and it is the problem in this one is there are many asymptomatic animals which will have LSDV. They may not show the exact skin lesions. They will have, however, they will have the virus. They may act as a source of infection to the other. Usually, the farmer treats or the veterinarian treats the animals which have a clinical signs. There are many cases of asymptomatic viremic cattle that causes the force for the epidemiological or control of the disease. And of course, this is not been that much proven that it can infect the human beings. Then when it comes to the clinical science, already sir has explained. I'll go to the some of the slides where we have undertaken field investigations to explain better. These are the local breeds of Karnataka. These are around 1 year old, both of them. Bullocks, future bullocks. It is a mild case in the sense you could not find the much lesions expected on the skin, but however they were having fever. They had only few nodules. This is one stage of the disease. Whereas if you look at this, here you'll find quite a more number of lesions as well as edema, and obviously the scapular or prescapular lymph node. It's almost you can hold with your full hand. That much enlargement will be there, and the developed edema will be there as well as front legs. Maximum edema will develop in the front legs, and sometimes they face even difficulty in walking. This is another case where the other side is prefusual edema. This was a breeding bullock which was infected. Once we told him, until that he was using for the natural service, so it is going to be a source of infection. The other side of the picture is in dry cow, which had multiple nodules of various sizes, including the this one anal region, vulval, all these things. That makes even artificial insemination or during the artificial insemination or during the natural service, your semen itself gets contaminated, or even veterinarian's hand will get contaminated, and you are going to insert LSTV. This is one more typical case where you can see what we observed are the things are, if the nodules size is bigger, number of nodules will be less. When the nodule size is small, you'll find numerous nodules. And what we observed, more local cattle getting disease when compared to the crossbred. However, when a crossbred gets the disease, it's more severe when compared to the local breeds. This is middle stage. This is the last stage of the disease where scabs are about to fall down. This was the case I was telling, sugarcane, this one. It can spread. This side is a 2-month-old calf, because the calves are sold once the moment it's a male calf. They usually sell up these crossbreeds. So they go to the market and spread the disease to other animals. These are the source of infections. This is one of the HF cattle, and the Jersey, as I said, if the nodule size is big, you see less number. When the nodule size is less, the left side one animal, it died after 3-4 days, because that farmer was practicing. If you are in the southern part of at least Karnataka, the farmers have the habit of giving a bath to a crossbred cow if it is a milching stage. Every day they give in the morning. Even if a farmer don't take bath, he will never forget to give a bath to his cow to get good milk. In spite of suffering, he started giving the bath. that get aggravated with a lot of secondary bacterial infection and dermatitis, including it has a cataract also. This cattle died. And the other form or key to what I was saying, they cause certain complications of mastitis also. Because if you look at this, both of them, this side is a heifer, this side was almost third stage or third lactation one, and a cataract. This will complicate with cataracts, usually complicate with maggot wounds. And many times LSD lesions are found in and around the eyelids. On the face are more, on the front side is more when compared to the back side in case of males. In case of females, back side is more when compared to the males. And the one of the first complication is maggot wound. If you look at from the distance, you may not able to see much. I just graphed in more magnifications. When you look at the white spot, then if you magnify, these are the eggs of the musca. Then it's going to be one fine day, the maggot will form that wound and complicate a lot of the case. Then as sir was already said, it has a seasonality, because the moment any disease which has a vector as its mode of transmission, it should have a seasonality. So obviously it depends on the vector population. So this was in one of the Turkey study over a period of 30 years with the temperature they plotted. Whenever the temperature was more, the population of vectors used to be more, the disease incidence or the prevalence used to be more. And this was again in one such case or study in case of Ethiopia. In India, we can take up such kind of studies, obviously, because still we have only 1 year outbreaks. We can collect the data and we can do this kind of work also to understand. But this already Sudhakar sir has said, just a glimpse of diagnosis. Until unless you don't diagnose, it is quite difficult to give vaccination or get a vaccine from the government. It's a simple virus detection. The PCR is the most, or 99% everybody recommends PCR. Then virus isolation, all these are of academic or research interest. Antibody detection is again, it is only when you don't have sample for either nucleic acid detection, or it is of post-vaccine testing, efficacy testings. And when it comes to the control, as I said, why government of India opted for goatpox vaccine? It is because of more relatedness, and this is the first time we got the vaccine, outbreak, and moreover, we don't have the local LSTV vaccine. Vaccination, if you look at that bar diagram, the one of the study, they said that first time vaccination, you may not find the titer. As you vaccinate 2 times or third time, the titer you are going to get is more. And the best way is obviously the vaccination, because it is a vector mediated. Vector control is going to be crucial for breaking the chain of transmission. And obviously you have to segregate as much as possible animals, and regular use of insect repellents, whether it is commercially available or we have know-hows of NPKs or this local knowledge practices, so traditional practices. So these can be used, like they do fumigate the sheds with neem leaves, putting and dried dung in the evenings. Such kind of practices are there. Sometimes they even apply aloe vera gel on the wounds. All these are there. And obviously, if you increase the farm level biosecurity practices, like once you get the disease, at least that animal should stay there. It should not be allowed to graze with the other sharing, or controlling the insects, or including a veterinarian attending that case. If you are attending the cases in your veterinary hospital, you try to attend or complete all the other cases. Then before you go to the home or before you leave the hospital, you can take up the cases of these LSTV, because it is also contagious or it can spread in that way. then use of disinfectants for cleaning sheds, because that's where the breeding grounds of your insects, whether it is mosquito or flies, and of course this ticks, and proper disposal of your carcasses. This diagram, it represents one of the modeling where stomaxis flies have been used to infect the cows. So the highest or peak it will achieve the infection rate, or 50% of your cattle out of 100 is going to infected around 27 days or 28 days.
Manjunath sir, your voice is not audible. Shashi.
Yes, sir. He's in mute condition, sir. Manjunath sir, can you
Sir,
please
sir,
unmute yourself?
Manjunath sir. Sir, please unmute yourself, sir. You are on mute mode, sir.
ಇವಾಗ ಆಯ್ತಾ? ಇವಾಗ ಕೇಳಿಸ್ತಾ
ಹಾ,
ಇದೆಯಾ?
ಈಗ ಕೇಳಿಸ್ತಾ ಇದೆ. ಅವಾಗ ಇದು ಮಾಡಿದ್ರಿ ನೀವು.
ಓಕೆ ಓಕೆ, ಇದು ಟಚ್ ಆಗಿರ್ಬೇಕು ಅನ್ಸುತ್ತೆ ಸರ್. ಲಾಸ್ಟ್ ಸ್ಲೈಡ್ ಅದು ಲಾಸ್ಟ್ ಈ ಸ್ಲೈಡ್ ಹೇಳಿದ್ನಲ್ಲ, ಅದು ಗೊತ್ತಾಯ್ತಲ್ಲ?
ಎಸ್ ಸರ್, ಯು ಹ್ಯಾವ್ ಫಿನಿಶ್ಡ್ ದಿಸ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಪ್ರಿವೆನ್ಷನ್. ನಾವು
being at NIVEDI, what we can do, or all of us can do is we can share the protocols and the methods what we have developed for diagnosis, or including the investigation and laboratory diagnosis. Any of your students or university or anywhere else, they can send some of the samples if your load is more, or if you have any students are running a projects, we are ready to collaborate. And of course human resource development and
to share by him during these deliberation may be useful to all participants. Thank you, sir. Over to Manjunath.
Thank you, Dr. Ravindra Biji sir, for giving a brief introduction of our beloved teacher, Dr. Vivek Kasaralikar sir. Now may I request Dr. Kasaralikar to go ahead with this topic, sir. That is therapeutic approaches for LSD. Over to you, sir. Sir, you are, sir, you are on mute mode, sir. You are on mute mode, sir. Unmute yourself, sir. Sir, please unmute. Sir, unmute, sir.
Yeah, Manjunath, can you listen?
Okay, sir. Yes, sir. Now you are audible, sir.
Okay. 1 minute.
Now you are.
Minute. Okay. Minimize करायचं. Why it is not minimizing?
काय minimize करायचं मला? एकाच ब्राउझर मध्ये तर minimize कसं होणार? दुसरं काय करायचं?
Close करायचं. 1 minute. 1 minute. मध्ये यायला लागलं.
इकडे बसा ना, इकडे या. कसं ते माझं
Manjunath, is it visible now? Manjunath?
Sir, It is visible now, sir.
Am I audible to everybody?
Yes, yes, sir. Yes, sir. It is audible.
Okay. So thank you very much. Let us start with the presentation. So earlier, two eminent speakers from National Institute ICR, they have spoken at length on the epidemiological aspects of lumpy skin disease and the diagnostic approaches. So my presentation in today's webinar is restricted on clinical presentation and therapeutic management. Now, let me make a brief, roundup about what has been discussed right now. So let me have one more small roundup for the people who have joined recently. So it is a lumpy skin disease, is a viral disease caused by a lumpy skin disease virus belonging to pox viridie, and it has got a very close identity with the Capri pox virus. Now, already OIE has given guidelines about the occurrence, then the presentation, and the therapeutic management, though they have given a broad guidelines, but we will be discussing in detail. So, as per the OIE guidelines, it is a notifiable disease with a morbidity rate reaching up to 50%, and mortality, though it is low, but in some of the cases, in some of the endemic areas, it may reach up to 5%. That's why it is of great importance, because it may cause a lot of loss to the farming community in the form of working bullocks or the productive animal. Of transmission, it must have been discussed earlier, but let me give a brief that arthropod-borne disease. So arthropod vectors like mosquitoes belonging to the genus Aedes and Culex, biting flies, especially the two genuses like Stromyces and Biomia, they are involved. And this is a very interesting, reference in the OIE guidelines and OIE LSD, whatever the brochure they have brought out. Male ticks, basically belonging to the Rhipicephalus and Amblyomma genus, they are the main transmitting agents for the disease. Now, this is a quite interesting topic for the budding veterinarians who want to work on the epidemiological aspect. And a lot has been, the work has been done to designate that the male ticks, they are the only one which are responsible for transmission of LSD. Now, transmission through artificial insemination and fomites has not been established. However, close contact with the infected animals always pose a risk of contracting the disease. Now, the disease is prevalent since 1998 in African countries, and later it has spread to the Middle East and Southeast Europe. Especially the first report goes from Egypt, and still the people are working, the scientific community in Egypt, they are working on this disease, and they are contributing a lot in understanding the lumpy skin disease. The first report in India, as Dr. Sudhakar must have told, or Dr. Manjunath Reddy must have told, that it is from Odisha. The date is also there, 12th of August 2019. The first report of LSD in India is reported from Odisha state. And then in OI, the reports, it is available. The government of India has intimated to the OIE that the first report was reported from Odisha. And now all of us, we are seeing the outbreaks occurring everywhere, almost in India, and especially in Karnataka. The disease is rampant, and it has been almost encountered in most of the districts. Now, fortunately, one more thing which we have to understand, the virus is highly host specific, and that's why the disease is being encountered in cattle and buffaloes, though the clinical form of the disease is less what you can call as severe in buffaloes. But we have report, and I will be sharing few photographs where the occurrence of LSD has been seen in buffaloes. Okay. Then OIE report indicates that the higher susceptibility of the disease is in crossbred animals. But now, in present scenario, what we are seeing is the indigenous cattle are also having the equal susceptibility, and the disease is also occurring in indigenous cattle. And the more concern with us is the severe form of the disease, which is very commonly encountered in young animals. I'll be sharing few photographs of the LSD in young animals, and especially the respiratory distress what the young animals they suffer. It is a very you can see how much of suffering is there in young animals. Okay. Then this point I want to highlight over here: the disease is not zoonotic. In WhatsApp and so many other social media, few reports, some of the photographs they are showing that the lesions are there on the hands of the people who were in closely contact with the LSD affected animal, like the abattoir workers and all. But still, the OIE manual says that the disease is not zoonotic. But anyway, the disease is new, so that's why in case it has got some sort of zoonotic importance, and in days to come, people will reveal that, and that's why the veterinarians working on this, the animals affected with LSD, they have to little bit be sure that they wear gloves and then they treat the animals, so that in case if any hidden form of zoonosis is there, that should not cause a concern in the people who are in close contact with the infected animal. The incubation period, experimentally, it has been proved beyond doubt that it ranges between 4 to 14 days. But the OIE manual says that it can extend up to 28 days under field condition. This point is very important because migrating animals, even 28 days, if you take it, the disease can appear, the clinical signs of the disease can appear even after 1 month after the exposure to the infected animal. So that is what we have to understand from this. Now let us go to the main important feature, that is clinical presentation. Now here I will be sharing the different clinical signs which are exhibited by the animals affected with lumpy skin disease. So fever ranging from 103 to 105 degrees Fahrenheit. Then the skin nodules, that is the most prominent clinical feature which is seen in almost all the animals, and that you can take it as a pathognomonic clinical sign of LSD. So most of the animals, they will have this skin nodules, and the size of the skin nodules can be from 2 to 5 centimeters, and they are very thick, extending into the deep layers. And sometimes these skin nodules, they coalesce or join together to form a larger size nodule also. And these larger nodules, they create some sort of difficult situation, because sometimes a necrotic area develops around the nodule, and then the whole nodule gets detached from the underlying tissue and gets sloughed off. So that has been mentioned over here. The sloughing of these nodules can also be seen, and there is a very slow healing of these lesions. Then the depressed appetite. The animal will be like taking less feed, or sometimes it will be totally anorectic also. Maybe inflammatory process. The animal is in a lot of pain. So because of that, the animal will exhibit some of the other clinical signs like depressed appetite, the less water intake and all. Enlarged lymph node in almost all the animals, because it has got the virus has got an affinity towards the cutaneous as well as the mesodermic tissues. So probably the virus multiplies in the lymph nodes. Because of that, especially in the lymphocytes and all. So because of that, the enlargement of lymph node is very distinctly seen in the animals affected with lumpy skin disease. Then this is another which we'll be discussing when I'll be showing you the photographs. Edematous tendencies. Okay, so the edema of dependent parts is very much marked in the animals affected with lumpy skin disease. And this edema, it varies depending upon the case. Sometimes it can be seen in only one limb, it can be seen only in the 4 limbs, or it can be seen even in the hind and forelimbs also. And especially the brisket, from dewlap to brisket, this edema can also be seen, and a few of the photographs I'll be sharing with you. So instead of discussing this theoretical chart, let us go to the clinical presentation photo-wise. So here you can see, as I already told you, the pyrexic reaction in acute cases of LSD is remarkable. Here you can see a digital thermometer which is inserted in the rectum is showing around 105.2 degrees Fahrenheit temperature. Okay. So then this is the skin nodules present on the young animal. Here you can see the whole body is covered with different size nodules. And especially, as I told you, in young animals, the respiratory infection is very prominent. Here you can see even the nodules, they are so big that it is even closing the eyes also. Here you can see this much of big nodule is there. The main problem in young cows is the respiratory form. And here you can see the plugging. The mucopurulent discharge, it plugs almost both the nostrils and creates so much of difficulty in inspiration and expiration. So because of that, even you can see the open mouth breathing also. And that's what we are following in clinics when I'll be talking about the treatment of the cases of LSD. This mucopurulent plug has to be removed every 6 to 8 hours. Okay, so you have to, with the help of cotton or with the help of stick, you keep on removing this so that the respiratory distress can be reduced in the affected animal to some extent. Okay. So here you can see one more calf. Different varying size of nodules are there all over the body. Some nodules, they are ruptured and created a wound over here. The whole body is covered with skin nodules, ranging from 2 to 5 centimeters. What I have already told you. Now, this I was telling you. Some of the nodules, they get necrotic. They develop a necrotic band around it, and then there is a sloughing of the whole nodular mass. It gets detached from the underlying tissue, and the whole mass gets sloughed off. So this type of sloughed wound can be sloughed off, nodule wound can be seen, and we are finding that the healing process in this type of wound is very, very slow, and it creates a lot of pain as well as discomfort to the animal. Okay. Adult animal photograph I have focused over here. Here you can see the larger nodules. Here you can see the smaller nodules. So the whole body is affected with nodular appearance on the body. So no part is exempted. You can see from head up to the tail. At the base of the tail also sometimes the nodules can be seen. This is what I was telling you. Among the cattle and buffaloes, we say that the disease is more prominent and more severe in case of cattle. But the buffaloes also, they get the lumpy skin disease, maybe in the milder form also. So here you can see the animal is presented, buffalo is presented with lumpy skin nodules over the body. Though you can compare the cattle, the appearance of nodules in cattle and appearance of nodules in buffalo. Here you can see they are scattered over a whole body. Here you can see a smaller nodule, right? Whereas in cattle, you have seen that the whole body is covered with so many nodular growth from whole body is covered. So this is what I want to do. The case which is presented to our hospital, I'm just showing you the photograph. So buffaloes are also susceptible, and we are getting lumpy skin disease in buffaloes also. Here one more buffalo. Here you can see a very large size, maybe about 5 centimeters or so. You see a large size nodule there. Here also one more nodule there. Okay. Then you can see the swelling near the prescapular lymph node. So like this, in buffaloes also, you are getting a typical nodular lesions in the buffaloes which are affected with lumpy skin disease. Now here, the edematous tendencies. I was telling you the edema of limbs, brisket, face. This is a downy bullock which was presented to our hospital, and here you can see the facial edema is also very prominent. Here you can see the rope which is used for control of animal. It has actually dipped into the the edematous swelling. Here you can see the typical notch is there, isn't it? So that is because of the swollen puffy face, and this has caused a furrow over here. And the whole body is covered with different sizes of nodules. And the edema, it is starting from duodenum, it is extending up to the brisket. Okay. So all 4 limbs are affected here. All 4 limbs, though you may not be able to appreciate it, but when we have seen the clinical case in our OPD, we have seen that there is a puffiness spreading from on the lower limbs also. So all 4 limbs, brisket, facial edema, this was seen in this case. So that's why we are sharing this photograph. Then here you see the hind limbs, though the lesions are there on the hind limbs and the whole body, but the edema is more prominent only in the 4 limbs as well as in brisket. So this is a some sort of application. The farmers, whatever it comes to their mind, maybe healthy paste or something like that, they have applied over here. So in a attempt to reduce this swelling, but here you can see a typical brisket edema is there, and the edematous swelling is starting from the scapular lesion going up to the patella area. Okay. Now this is a very distinct edematous tendency case. Here you can see so much of prominent brisket edema is there, starting even the submandibular edema is there. Now let me tell you, just take out your discussion on LSD. If such kind of case is presented in hospital, most of the veterinarians who have joined today's discussion. So in most of the pericarditis cases, you get this type of brisket edema, and it is, as a matter of fact, it is the outcome of congestive heart failure. So if you take out your knowledge about the LSD, if such kind of case is presented to your hospital, in 80 to 90% of the cases, you will presume that it may be a case of pericarditis or congestive heart failure leading to brisket edema. You observe these are the lumpy skin nodules scattered over the body. So it is a complication of LSD, and that's why brisket edema is there. Okay, and we have seen the case recovering over a period of time with the routine treatment with diuretics, antibiotics, and anti-inflammatory. No, this one interesting article came across when I was searching the different publications on lumpy skin disease. This is a article published in Alexandria Journal of Veterinary Sciences, and Mahmood and co-workers, they have reported the clinico-biochemical and electrocardiographic changes in cattle naturally affected with lumpy skin disease. This actually article is focusing, and it is giving a more elaborate picture that there could be a illustrious, or there could be a effect of LSD virus on the heart. Okay, so there may be a cardiac involvement in LSD, and this has been typically they have recorded that there is a conduction defect in the animals affected with LSD. You can see the abdominal P wave, especially the P wave amplitude is reduced. The T wave amplitude is also reduced. So the P wave amplitude, it tells you something about depolarization of the auricles. T wave indicates something about the depolarization of the ventricles. So something is going on. Okay, so because of that, the amplitude of these two important markers of PQRST, it is changing. So lower amplitude of P and T wave has been consistently recorded in the animals affected with lumpy skin disease. So this article probably it will open new era for more research whether there is a cardiac involvement and whether there is any specific marker. Even they have tried to measure the cardiac troponin 1, and they said that in few animals elevated levels of cardiac troponin also has been observed. So probably there may be a myocardial involvement in the animals affected with. Then there is a decreased RT interval. So this has been documented, and this opens a new area for the research in the animals affected with lumpy skin disease. And at our college, we are already working on that. 3 students, our postgraduate students, they are working on different aspects of lumpy skin disease, and few of them probably come up through their research to throw some more light on the lumpy skin disease. Then this respiratory disease I was telling you. So the same cough, you see here, you can see how much of respiratory difficulty. The open mouth breathing. The animal is having gasping type of breath. This is another side photograph of the same animal. Here you can see during each respiratory cycle, even there is a closing of eye, because there is a lot of effort the animal has to make to do the inspiration as well as expiration. And in such cases, in treatment, we will be talking about nebulization has helped greatly in reducing the respiratory distress. Probably the nebulization must be helping the bronchodilation and thereby helping the other agents to reach to the pulmonary parenchyma and exerts its effect like antibiotic, anti-inflammatory and all. Okay, so this is a very typical, and as I told you, the respiratory distress and respiratory form is very commonly encountered in young animals. Now, these are also we want to share with the audience that ocular lesions, whether we can call it as ocular form or something like that, but there are a few cases where we have recorded typical lesions in the eye, especially on the conjunctival lesion. So here you can see a typical nodule is there on the corneoscleral junction is there. Here bulbar as well as scleral conjunctiva. Here you can see there is a small nodule is there. Okay. Here you can see the bulb, especially corneoscleral junction. You can see a typical nodule involving the eye. So LSD virus can cause such type of lesions also. This one more photograph we wanted to share with you. Here you can see the corneal opacity. The virus is responsible for opacity of cornea, or there is a secondary bacterial complication leading to corneal opacity. We have to ascertain that, but such type of things can also be seen. You can see the whole body is affected, and you can see the skin nodules. Along with that, you can see even the corneal opacity also. It can be unilateral, it can be bilateral also. Okay, so this is what the ocular lesions of LSD. Then the enlargement of lymph nodes. Here you can see the lesions of LSD are there, but they are almost healing. But here you can see with digital pressure, the prescapular lymph node has been focused. So here you can see almost 3 to 4 finger enlargement is there. Okay, so the enlargement of lymph node is also an important clinical finding in the animals affected with lumpy skin disease. Now, coming down to the another important aspect of my talk is therapeutic management. Now, let me tell you that there are no specific guidelines available in OIE manual how the disease should be treated. Only under the brief headings like broad spectrum antibiotic, then the anti-inflammatory, then wherever edema is there, the diuretics and other things they are being used for the treatment of lumpy skin disease. So whatever I am sharing in my presentation is our personal experience by looking at the effect of different antibiotics in our clinic. So what we are using that I am sharing with you, and we are getting a very good response to this. Okay, so the first preference, if an adult animal is affected with lumpy skin disease and both the skin lesions are there, and you have to categorize them whether it is acute form or a little bit of chronic form. Okay, so if it is acute form, what we are following is we are giving enrofloxacin at the rate of 3 to 5 milligram per kg body weight in acute cases for about 3 to 5 days. Then if the case is chronic, the lesions are already healing, the enrofloxacin long acting can be given at the rate of 10 milligram per kg body weight once in 3 days, about 2 to 3 times depending upon the response. Even we have used in few cases the marbofloxacin, which has been which has come into the market recently, and it is a wonderful drug in fluoroquinolones. So marbofloxacin, it has got 2 schedules. If it has to be used every day, the dose has been reported to be 2 milligram per kg body weight for about 3 to 5 days. If you want to give on a higher dose, maybe from 6 to 8 milligram per kg body weight, then the single dose of marbofloxacin is enough. So we have tried both the things, and equally we are getting a good response to marbofloxacin also. So these are the first priority antibiotics which we are using in our clinic for the treatment of lumpy skin disease. Now, what I am telling is, in case of young animals, there is acute respiratory form, and in such type of cases, our preference of antibiotic is ceftriaxone. Either it can be used alone or along with the tazobactam at the rate of 10 milligram per kg body weight for about 3 to 5 days. Okay, so what we are following, let me tell you, if the severe respiratory distress and other things are there, we give first 1 or 2 days intravenously the ceftriaxone alone or ceftriaxone with tazobactam, and if the case improves over after 1 or 2 days, then we go for the intramuscular injection. Even we have given enrofloxacin in few cases of lumpy skin disease by intravenous route. Only thing is you have to read the label. Only few companies they say that their enrofloxacin can be used intravenously. Okay, so keep in mind with the product brochure should be available with you. If the company says that the intravenous injection of enrofloxacin is safe, even it can be given by the intravenous. Okay? So this is what. Now let me talk about the cost effectiveness of the treatment. As far as the large animal practice is concerned, we have to give emphasis to the cost effectiveness also. So sometimes if you have your dispensary is having antibiotic with you, you can use your antibiotic. But sometimes if the antibiotic has to be prescribed, in such type of situation, you can go even for the cheaper alternatives like streptopenicillin. So sometimes we are also using streptopenicillin and 2.5 gram all these days we were using, but we are finding that it is underdosing. So nowadays we are following 5 grams daily for about 3 to 5 days. So this is what I wanted to stress upon the use of antibiotic in the treatment of lumpy skin disease. Let us go to the supportive treatment. What else can be given? Okay. Pain management is the topmost priority in the treatment of lumpy skin disease, and NSAIDs, that is non-steroidal anti-inflammatory drugs, they form the front line in pain management. And the choice, it depends upon the veterinarian. Here also, we normally use in mild cases the meloxicam alone at the 1 milligram per kg body weight. Otherwise, if pyrexic reaction is too much beyond 103, 104 degrees Fahrenheit, then we use preferably the combination of meloxicam as well as paracetamol. The other alternatives which are available are tolfenamic acid derivatives, and it has been, the pharmaceutical background of tolfenamic acid says that it stays there for a longer period of time in the body. So that's why it gives a anti-inflammatory effect for a longer duration also. So it can be given at 2 milligrams per kg body weight. Then we have a newly introduced flunixin-meglumine, and it is a wonderful non-steroidal anti-inflammatory drug. Especially the flunixin has proven its efficacy in equine colic, and here also we are finding the flunixin in severe cases of lumpy skin disease at the rate of 1.1 or even 2.2 milligram per kg body weight, gives wonderful effect as a pain management strategy. Okay, then the antihistamines also should be there in the protocol of supportive treatment of lumpy skin disease. Either phentermine malate or chlorphentermine malate can be given at a recommended doses. So that will reduce the histamine reaction at the tissues, especially this skin form and other things. So there, the antihistaminic effect with the help of these drugs can be exerted, and you can reduce the irritation and itching sensation at the skin lesions. Then along with that, supportive treatment also should be focused on supplementation of fluid and electrolytes, because the animal is not taking the requisite amount of food, it is not drinking the requisite quantity of water. So dehydration, electrolyte and energy imbalance is likely to occur. So you can use the isotonic fluid should be preferred because the dehydration is there. Even if you feel that the animal is not that much dehydrated, even you can go with the hypotonic fluids like Rintos and other hypertonic solutions, including and percent glucose solution also. Now, this is what I wanted to discuss at length with you people. Now, when a new disease strikes, okay, the scientific community, they keep on overthinking about it. So the COVID-19 especially, you must have seen, you must have read the reports that out-of-box thinking is required for the treatment of newer diseases. So LSD was not existent in India before 2019. The first outbreak was reported in 2019. So it is hardly, you can say, 1 year experience is there with us. And it will, you have to explore the different strategies which will help in the treatment of lumpy skin disease. Like in COVID-19 also, you must have seen that hydroxychloroquine is there. Then even ivermectin has been told to give a protective response against the COVID-19. And most of the health workers, they are taking ivermectin as a prophylactic dose, and it has been proved to be effective to act as a protective, or it gives a prophylaxis against COVID-19. So here also, though the reports are scanty about the use of different drugs, so we have a thinking that vitamin C, being an antioxidant, which may give protection from the free radical injury to the tissues. It also, vitamin C also has been documented to promote the tissue healing, which is very much required in lumpy skin disease. And apart from that, vitamin C, the role of vitamin C as a cardioprotective drug also has been well established. So all in these three fronts, we thought that the vitamin C should be used in the cases affected with lumpy skin disease, and we are using so 1000 milligram, almost, theoretically they say that up to 3 grams, means 3000 milligram per day, vitamin C can be given. But we have very few market preparations which has got a higher concentration of vitamin C. We are using a commercial preparation available with us, that Dorbic. It has got about 250 milligrams of ascorbic acid, that is vitamin C per ml. So we are using in normal lumpy skin disease cases up to 1 gram, that is 1000 milligram, and in severe cases even we have given up to 3 grams, that is 3000 milligram per day for about almost for a week or so. Even you must have seen in COVID, they say the 1000 milligram vitamin C should be taken for about a month or so. So it is a vitamin C, it is a daily requirement, and it doesn't have any toxic effect on the body. So this can be given for about a week to 10 days or so. So our feeling, we are getting we don't have a direct documentation. Maybe some of the PG work students who are working on the therapeutic management of LSD, once their thesis comes out, probably we will be able to focus more light on the response of vitamin C in lumpy skin disease. So likewise, out of box thinking, what we are thinking is immunomodulation is very much required, because it is a viral disease. Okay, so for viral diseases, nothing helps other than the immunity. So if you have certain drugs which will be immunopotentiating effect or immunomodulating effect, such drugs should be tried in the treatment of lumpy skin disease. And so here we are using levamisole at the rate of 2.5 milligram per kg body weight. The therapeutic dose of levamisole is 7.5 as a anthelmintic. Here we are using subtherapeutic dose as immunomodulating, which is already established. 2.5 milligram per kg body weight. Levamisole injectable is available, can be used. So it has been well documented that it improves the cell-mediated immune response, which may be required to get a protection or some sort of immune response against the lumpy skin disease. So that's why we promote that the levamisole should also be given a thinking in the treatment of lumpy skin disease. Now, one more, the aspect is the role of alternative medicine. As I know, and I substantiate also that it is a quite debatable type of topic, because veterinarians, they do not have a direct exposure on the knowledge of either Ayurveda or homeopathy. But I am a executive committee member of Indian Society for Veterinary Medicine. I am a joint general secretary of the society. So we conduct regular meetings, and then every year we have a conference. So in last conference, we have taken a decision that there should be a boost for the use of alternative medicine, because that is very much required. And no science is a fake science or something like that. So unless until you use it, you see the effect of alternative medicine, you cannot outrightly say that the Ayurveda should not have a place in the treatment of. And few veterinarians, they are actually working on this Ayurvedic as well as homeopathic treatment of animals, and they are reporting that it has got a better effect in the treatment of lumpy skin disease. So Pashudan Prahari is a journal which promotes the scientific report writing. And in published Pashudan Prahari, this is how they say that these are the different Ayurvedic products. The Guduchi, Gulwel, the scientific name for this Gulwel is Dinospora cordifolia. So it should be taken as 5 grams. Adrak, which is routinely available. Gingiber, officinate, 10 grams. Turmeric powder, which has got an active ingredient of curcumin, 10 grams. Neem leaves. Okay, neem leaves also. There is a publication from Ayush department. So they say that neem leaves has got a antiviral properties, and especially in COVID cases, they say that this neem leaves should be boiled. Either a little dose of this decoction can be taken orally, but if you take a bath of neem leaf decoction, probably it also exerts anti-viral properties, and it gives some protection to the people as a prophylaxis. Okay, so here also the report says that the neem leaves, 1 handful of neem leaves can be taken. Betel leaves, which you use as a routinely to take 1 and all. So 5 numbers. Garlic pearls 2 numbers. Coriander and cumin 10 grams. Black pepper 10 grams. Bay leaves 10 grams. Jaggery 100 grams. Okay, so in 100 grams of jaggery, all these things should be made into a paste like thing. Make 2 doses, and these 2 doses can be given at an interval of about 8 hours, morning and evening, for about 5 to 7 days. And there are reports that it gives a relief in the cases of lumpy skin disease. Likewise, homeopathy also, there are variety of homeopathic drugs which are being used for LSD. So I'm not going in detail about the different formulations of homeopathic medicine, but this is a product which is one Goel Pharmaceutical is there in northern India, and they are marketing this LSD 25 kit. So the photograph says that LSD 25 kit. It has got 2 bottles. One is for external application. 1 bottle contains a formulation which can be given orally. So they say that 20 to 25 drops can be given orally, the oral formulation, and the other bottle.
Hello, sir. Sir, it is not in slide mode, sir. Presentation mode. Hello. Shashikiran.
Yes, sir. Sir is there, sir, but he's not able
Voice
to
is
speak.
not audible.
Yes, sir. His video is stuck, sir. Like it's better that he can join. Definitely, you can continue, sir.
I think he has left. He will join again.
He will join, sir. He will join. Sorry for the inconvenience, participants. Definitely, the sir will be joining soon. So after the presentation is over, we're gonna share the feedback form in the chat box itself. So you all can give your feedback form. Like you can give your feedback in the clicking that link. So the person who will be filling the feedback form will get the certificates. So for the next day, you will get the different feedback form as well as different certificates of tomorrow's also. Tomorrow also we have a great speaker.
Contact Kasaralikar sir to
Yes.
get in. You call, sir. Sir is online.
Manjunath?
Yes, sir. Yes, sir.
Sorry,
You are audible now, sir.
there was.
You can now share your slide, sir.
Yeah.
You can share your slide, sir.
Yeah. Sorry for the inconvenience. Manjunath?
Sir, keep it in slide mode. Show, sir.
Yeah. I'm going to the requisite slide. Okay. This was almost a new last slide.
Yes, sir.
But
Yes,
anyway,
yes.
yeah. So what
Right,
I was
right.
discussing was, so this is a market preparation available from Goel Pharmaceuticals, and it has got two components. One is oral medication, the other one is topical application. So a topical application comes in the form of spray that can be sprayed on the lesions on the skin, whereas the oral formulation, about 25 drops 3 times a day, can be given orally. And some of the people, they say that it is showing quite encouraging results. Now, this is what the therapeutic management we have discussed. One thing which the earlier slide, where in the supportive treatment, wherever the edematous tendencies are there, here normally the diuretics should be a part of your therapeutic regimen, and in many cases we have seen that the case response in about 3 to 5 days. Okay, if the extensive edema is there, it takes a little bit of time to reduce, but definitely the case gets the case responds to the diuretic routinely available. So there is a little bit of two schools of thought why there is edematous tendency in case of lumpy skin disease. Now we say that there is a lymphatic compression whenever the inflammation is there associated with lumpy skin disease. So probably there is a lymphatic obstruction or lymphatic system compression. So because of that, the fluid escapes and then accumulates into the dependent parts. That's why brisket edema, then edema of limbs is seen. But maybe few other people, they will work extensively on this. And sometimes what I feel is that as we have already a report which says that there is a cardiac involvement, whether this cardiac involvement is responsible for the edematous tendency, it is the days to come will tell you something about this. Okay, so in that type of situation, probably you may have to think about the cardiotonics and all. Okay, so time being, there are no supportive documents to say that directly whether you should try to give any cardiotonic. Time being, we are depending on the diuretics, and in majority of the cases, in about 3 to 5 days, you reduce the edema to a very great extent. Okay, so this last slide, that is a future scope for the treatment of lumpy skin disease. As I already told you, the disease is new to us, and whatever information is reaching from different corners will be a eye-opener for everybody, and then probably it will contribute further in improving the treatment of lumpy skin disease. So this is one more topic which we have to keep it in mind: use of antiviral antibiotic. I know the cost is a major factor in the treatment of large animal practice, but still, some of the, at least on experimental basis, if somebody works on this antiviral antibiotics in lumpy skin disease, probably it may give a new hope for the treatment of lumpy skin disease. Newer non-steroidal anti-inflammatory drugs, they are being pumped into the market. So probably few new NSAIDs may also come to our help for the treatment of lumpy skin disease. Then these steroids, the use of steroids, always we have apprehension because it causes a lot of immunosuppression. It has got its own toxic, what you can call side effects also. So there is, unless until it is a life-threatening type of situation, no veterinarian will give a priority for the use of steroids. But COVID-19 has taught us a new lesson. So steroids are the primary concept in the treatment of COVID-19, severe COVID-19 patients. So here also we have to give a thinking whether low dose steroids can be of some help in the treatment of lumpy skin disease. And new antioxidants. Antioxidants, I have already told you, and we have discussed about the vitamin C. Few reports may be there available for use of vitamin C. Likewise, we have got vitamin E also, selenium also, which also has got antioxidant effect. So probably some more reports may throw light on the use of vitamin E and selenium in the treatment of lumpy skin disease. These are the future scopes for the treatment of lumpy skin disease. Coming down to the prevention and control. Prevention and control, it is a two-pronged strategy. One is vaccination. Now, let me tell you that the vaccine, Lumpy Vax, which is globally available, it is not available in India. So that's why we have to depend on our other alternative. So we are even the advisory from ICR says, and Government of India says that the goat pox vaccine should be used for the as a prophylactic measure for the lumpy skin disease. So the goat pox vaccine Uttarakashi strain is used at the rate of 2 ml for large animals, 1 ml for the calves, and can be given to all the in contact animals. Okay, and in absence of goat pox vaccine, even it has been indicated that the sheep pox vaccine, because of its relativity with the lumpy skin disease, can be used as a prophylactic measure. The second important aspect of prevention and control is vector control strategies, because we have understood that the lumpy skin disease is an arthropod-borne disease. So if you control the vector effectively, probably you will help in reducing the outbreak and the spread of lumpy skin disease. So here, the ectoparasites, which we have already seen, like especially the ticks, we can use ivermectin for the treatment, as well as there are variety of ectoparasiticides which are available that can be applied to take care of ectoparasites. The fly control strategies that plays a very important role, because biting flies, they are the major source of the virus transmission. So in fly control strategies, either you can go with the insecticide spray in the barns or where the animals are tied, the animal houses. Then the most important thing is the proper disposal of farm manure and waste, because this farm manure, wherever the dampness is there, manure is being stored. So these are the breeding grounds for the flies, biting flies especially. Okay, so this should be either covered with a net or a proper disposal should be done, and wherever the manure is being stored, it should be covered so that it will not create a breeding ground for the flies. So that should be kept in mind. And even application of neem oil. Neem oil itself is a insect repellent, so it can also be used wherever the animal sheds, they are facing the fly menace and the mosquito menace. So the neem oil can effectively act as a insect repellent, and to some extent it can reduce the ectoparasitic bites to the animal, and thereby may be playing a role in prevention of lumpy skin disease. Okay, so this is what I wanted to share with all of you. Thank you very much, and I want to take this opportunity to thank especially the International International Veterinary Students Association Bidar, which has given an opportunity to have a national level webinar where we can discuss at length about the different aspects of lumpy skin disease. I am also thankful to the Alumni Association, which is also a part of organizing team for today's 37th College Foundation Day of Veterinary College Bidar. And also I am thankful to Dean Veterinary College Bidar, Dr. Dilip Kumar, for identifying me as one of the faculty for sharing the information on lumpy skin disease. Thank you one and all.
Thank you, sir. Thank you. Am I audible? Thank you, sir. Thank you for your wonderful talk.
It's not opening. I don't know.
So just click
So
on the
if
stop.
I am audible,
Yeah, you're audible, sir. Just click on
stop.
sharing, sir. Yes, sir. It's stopped.
Right.
Yes, sir.
So yeah, I give the
Thank you, sir. Thank you for
where if any discussion or if any queries are there, I'm open to have a discussion on that.
Yes, sir. Yes, sir.
Dr. Manjunath is also there.
Yes, sir. Yes, sir. Yes, sir. Thank you, sir. Thank you for. Yes, sir.
Hope the presentation.
Thank you for a wonderful talk, especially on therapeutic aspects of.
Dr.
Yes, sir.
Seshupur is
Yes, sir.
also there.
Definitely. Many other. Yes, sir. Yes, sir. Sir is there, sir. Sir, thank you, sir, for a wonderful talk on the therapeutic aspects of LSD. Now, I request Ms. Puha to take up question answer session.
Yes, sir. First of all, I would like to thank all the scientists that have come and shared such valuable information with us. Thank you so much, sir. Our participants have some questions I would like.
Okay.
The first question is like how to differentiate LSD skin eruptions and allergic skin eruptions. How can we differentiate and how to distinguish?
Yes, Spro, it's a good question. Okay, I'm answering your question. Hello, am I audible?
Yes, sir, you're audible.
Yeah. So the what she has asked is the allergic nodules. How to differentiate between allergic reaction nodules and the lumpy skin nodules. Lumpy skin nodules, they are very thick, deep. Okay, so when you hold that nodule, you find a very hard mass. Whereas we call it as allergic, that those are urticarial lesions. Okay, urticaria. So they are called as dollar spots. Okay, so this dollar spot will have a elevated margin and depression at the center. It is a small edematous swelling. Okay, whereas the nodule of LSD is very firm, hard to touch. Okay, whereas allergic eruption has got a only elevated border. At the circular end, you find the elevated area. At the center, it is depressed. Okay, and if you just press the allergic nodule, it gets pitting. You can call it as pitting on pressure because it is edema, small edema. Okay, whereas lumpy skin disease, you have got a very hard nodule dipping, going up to even dermis level. Okay, so that is the basic difference. Very simple.
Pain will be more, sir, in lumpy skin. In allergy, pain will not be there, sir. Animal will not.
Yes, sir. Yes,
pain, no, sir, pain. In lumpy skin, pain will be more, sir. ಜಾಸ್ತಿ ಇರುತ್ತೆ. ಅಲರ್ಜಿಯಲ್ಲಿ ಟಚ್ ಮಾಡಿದ್ರು ಕೂಡ pain ಅದು animal reaction ಕಡಿಮೆ ಇರುತ್ತೆ, sir.
Thank you so much, sir. One more question is like the Panchangavya mixture of go, mutra, ghee, butter, dung, urine, and saliva, does it help in the treatment of LSD?
No, I don't have right now available report with me, but Panchagavya, people are working on that, and it has got its own therapeutic effect. So probably maybe having some healing effect in LSD also, but I have not come across with any available report.
Okay, sir. Thank you, sir. One more question. What are the hematological and biochemical changes in various stages of LSD?
Dr. Boyer? If the screen is shared, okay, 1 minute. Mallikarjun?
Hello.
Sorry, Manjunath.
Sir, Manjunath Reddy, sir. Can you?
Kelsa.
No, there is an article. I just focused on Mahmood et al.
Yes, sir.
So it has got some. I will share. You can send me the email. I will share the report of Mahmood et al. From Egypt. So they have studied the different aspects of LSD, biochemical changes. There are so many parameters they have listed, which are changing. So here normally the lymph.
Lymphocyte adirattu sir. Neutrophilia irutte lymphocyte.
No, as a matter of fact, there should be a lymphopenia should be there, indicative of viral disease, but it doesn't, because by the time the case is presented, already the secondary bacterial infection takes upper hand. So you have got neutrophilic, okay, and a typical response, okay.
Any question?
What is that, Manjunath? Anything else?
Should I repeat the question, sir? Was it not? Yeah, I'll repeat the question, sir. What is the reason that the occurrence of LSD in buffaloes is low when compared to that of cattle?
Occurrence of LSD in buffalo
is low when compared to cattle.
So
your voice is not audible.
Yeah, okay, sir. Okay, sir. I'll talk louder. Sir, you can continue, Kasaralikar, sir.
True. No. You want a substantiation for that or what?
They just wanted to know what is the reason, sir. Any particular reason?
Maybe the thick skin, probably. The skin response is less, and maybe the whiting flies or whatever. I think epidemiological aspect, the other things they can, or maybe species resistance or something like that.
Sir, one is the probability of insect bite. You could kill buffalo in the same farm. Cattle is also there. The flies have the tendency to attack the cattle more than buffalo. And as usual, anatomy of the skin, the thickness of the skin is the second factor. And the buffaloes have that property of wallowing or that one that will cause the insects not to get touched or attacked much. These are some of the reasons. Still, definitely there is a need of research in that.
So I want to continue with the QA session. So what are the best methods of tool in field conditions, sir?
Can you repeat the question?
Yeah, it will be a good question for Sudhakar, sir. What are best methods of early diagnostic tool in the field conditions?
So early diagnostic tools for field conditions so far not available, but the clinical signs are very much characteristic in this LSD disease. So seeing the clinical signs, whatever we have been dealt so far for all the three speakers, that can be observed properly, and based on the epidemiological feature also can be accommodated in the diagnosis of the disease. So these clinical symptoms only can be observed in the field condition, and the diagnostic tools per se is not available for field purpose, and that is it for exactly.
Manjunath sir and Vivek sir, can you add something for this?
Yeah, in the field, as sir said, if clinical history is
and Dr. Sashibhushan has very correctly pointed out, very rarely you get a confusion in the diagnosis by looking at the case. Okay, very rarely, because the case is presented in such a typical form. Skin nodules, in about 90% of the cases, skin nodules are present. If you see the size of the nodules, the thickness of the nodule, I don't think any veterinarian should have a confusion in. No doubt you are talking about diagnostic tool, but the visual inspection of animal itself will add in the diagnosis of LSD. Now, let me share one with the permission of Shashibhushan and Manjunath Reddy.
Please.
The similar case we have recorded, the similar case, the one calf was presented with nodules all over the body. Okay. And as I told you, some of the PG students are working on LSD. So it was presumed that it is a LSD case only. Okay. I just, when I saw the case, I just told them, you just rule out cutaneous form of thyleriasis. Okay. And surprisingly, the calf was highly positive for thyleriasis. Okay. So this cutaneous form of thyleriasis is very rare form, and sometimes it can be a confusion in LSD. So that is what we have to see. So some, maybe Dr. Sudhakar, Sashibhushan, all of them, they are working hard to have a newer diagnostic tool. Maybe the days are not far when you will have a lateral flow diagnostic kit for the diagnosis of lumpy skin disease. Am I correct, Dr. Sashibhushan?
Yes, sure.
Yeah.
Sure.
So that will be a rapid diagnostic tool. Maybe we say that the viral load is very high in the cutaneous lesions. So maybe a scrapping or something that a sample can be a source of virus, and that can be put across a lateral flow diagnostic kit, and maybe the specific antibodies can be tagged to the device so that you can have a within no time you will be able to diagnose the lumpy skin disease. As I told you, the disease is very new to India. It's hardly 1 year. Okay, last August. Okay, so that's why a lot of work has to be done, and people are working on that, especially the two national institutes, NIVEDI and NISHAD. They are working hard to throw some more light on the epidemiological as well as diagnostic approaches for lumpy skin disease. So maybe in future you will have a diagnostic kit, which we call it as pen-side diagnostic, isn't it? Just besides the animal. Collect some fluid, put it in the lateral flow, and see in about 5 to 10 minutes whether it reacts or not. Okay.
Thank you, sir.
Right.
Any questions? Any more questions?
Yeah, even though, yes, sir, I will need to ask some more. Even though LSD first reported in 1929 for the first time, why we couldn't control outbreaks even in 2020, nearly 100 years? What are the practical difficulties?
No, in 1929 it was detected in African countries. Now it is in India. I consider you are concentrating on Indian scenario. It has come recently, and in several countries in Middle East also, they have eradicated the disease. So there is no problem in controlling the disease. Only commitment and a strong resources should be there. In India, we don't have the homologous vaccine. However, homologous vaccine in other countries are available, but it depends on the government policy whether to import those vaccines, because those are different strain. Again, in India, we will introduce that strain, that is live virus. And it has also been recently reported that there is a chances of recombination from the live virus to the vaccine virus, live field virus to the vaccine virus. So there is chance of evolution of a new LSD virus in future. In India, we have goatpox vaccine and shitpox vaccine, indigenous goatpox and shitpox vaccine that give the cross protection against LSD. So we are using that. Only thing why we have not controlled how much animal population so far has been vaccinated with goatpox vaccine, that also we have to look into. And at the field level, everybody knows the practical difficulties that has to be sorted out. Suppose in any village that outbreaks happens, whether the farmer is ready to report immediately to the local veterinarians, and whether the local veterinarians are ready to take the action against that outbreak to stop the further spread of the disease. So it's a responsibility of all the stakeholders for a commitment to restrict the further spread of the disease. I told in my presentation also, it is only 16 months when the first outbreak was reported, and now it is 16 states. So average every month, if we can take the data of average incursion of the disease in a state, so every month a new state is being reported for the outbreak of the disease. So the neighboring states, suppose there is already reported incidence of LSD, so the free states has to give importance for the prevention and control and no incursion in that state. So it is not like that we don't have the resource. We have the resources, but we have to put at one platform. It can't be controlled by one state only. It should be for control. We should have a regional approach for a nation to control the disease. If we are taking the surrounding neighboring countries, Bangladesh is already affected. Now Nepal, Bhutan, everyone is affected surrounding India, barring Pakistan. So, and you know that India, Bangladesh, Nepal, and Bhutan, there are lots of, we can tell that uncontrolled movement of even human or animals. So, at that point, we need to focus, and a strong commitment is needed from all the stakeholders, from the top official to the field veterinarians to the livestock owner. Then only we will be able to control the disease, because it is very new disease. Only 16 states or whatever 17 states are affected, so we can have all the opportunity to stop the further spread of the disease. But you have to help yourself, and you have to help your farmer, you have to help your neighbor. Then only we can control this disease. Otherwise, it will be rapidly spreading. It has a transboundary spread capability. So we can help ourselves only. Nobody can help us.
Thank you so much, sir, for the elaborative explanation on this. Sir, it's a last question I want to ask is, which vaccine, that is LSD vaccine or the goatpox vaccine, which is the most promising one?
No, see, in Indian scenario, if you are talking, then goatpox vaccine is very much available in India. LSD vaccine, homologous LSD vaccine is not available in India. So we have no choice rather than to use goatpox vaccine, or chickenpox vaccine also available. However, it has been reported that heterologous vaccine doesn't give the 100% protection in all instances, but it has the equal efficacy to protect cattle as good as homologous LSD vaccines. But certainly, for the eradication purpose, we need to have homologous LSD vaccine. And in future, very soon, we will have our indigenous vaccine in India. But till then, our government policy changes to import the homologous vaccine from other countries. Till then, we have to have a religious focus to vaccinate as much as possible the healthy cattle population surrounding the outbreak areas. Yeah.
Thank you, sir.
Just 1 second.
Please.
Yeah. To answer your question of why we could not control, see, stigmatization of diseases, mortality. Could you hear?
Yes, yes. Go ahead.
Any government or policy maker will look into what is the impact of the disease. The mortality is the first factor to be considered as the parameter to implement any control program. Example is FMD or avian influenza. Okay, LSD does not cause that much mortality when compared to morbidity. So that is why the policy, or it is going at a tortoise speed in the sense. So as far as vaccine, definitely the present is goatpox. You go about it, it can protect many things. There is no need of thinking too much ahead until we get the homologous vaccine. Could you get the answer?
Yes, sir.
Simple way?
Yes, sir. Yes, sir.
Yes, sir.
Any more questions, Shashikiran?
Yeah, last one, sir. It will be last one. Does gobar help in controlling this disease?
Gobar, let me have one.
It's a quite good question, actually.
Oh, you are talking about gobar.
This is question from Justin Deep Singh.
Yeah, I understand. Gobar as such, like that cattle defecated, and you are using that gobar. So, since this disease is vector-borne, and gobar and dung hills are very much good breeding sites for these arthropod vectors. So you can understand gobar may not help for for even restricting the spread of the
Sudhakar
disease.
sir,
Sir,
student, I like to ask student back the question: what form of gobar you want to use? There are two forms. Wet form is danger. Dry form is good.
okay.
The student, could you get back to me? What form of gobar you want to use? If you want to use dry form, if you put fumigation for that dry form, definitely it has an impact. Put the wet form, it is going to precipitate the condition.
Sir, he is asking dry gobar can be used.
Dry.
Yes, it is a good one to fumigation or as this one, no, shed disinfectant. It has a good effect.
To control the vectors.
My question. Yes, wet or dry, that is more important. Because some people it is not a gobar, it is called as a diamond.
Any activity which reduces the vector population in and around your cattle or your animal house, that will help to reduce the exploitation of the disease, definitely.
Any other questions?
Yeah, sir. We are running out of time, but
So there is
if you
one more question from Dr. David.
Two questions, sir. Does autoimmune therapy helps in the lumpy skin
David
disease?
Tilson. 1 minute only.
What is that? Repeat question.
Yes, sir. Like he's asking, does autoimmune therapy helps in the lumpy skin disease?
Does autoimmune therapy helps in LSD?
It is the disease.
No, the time only is the answer.
Sir, can I answer, sir? The disease is not a humoral immune mediated disease. It is maximum is cell mediated, plays a major role.
Yes.
So if you give your autoimmune or antibodies or serum, it's not going to have much effect because its maximum is cell mediated immunity, which plays the major role in pathogenesis also. Question?
No, sir. Like it's already a lot more. We had a discussion. If anything, the questions comes, definitely we'll approach
more
you.
Questions?
Please.
Yeah, we'll approach you
If
with
there
a
are no
email ID.
questions, okay.
Yeah, you can share the email IDs of the speakers so that.
Yes, sir. Like we will share the email IDs. If something they want to clear with you, definitely they can approach you, sir.
Definitely.
Please. You can share them with mobile also. Let them do WhatsApp also.
So I will say that one take-home message to participants: whenever suspicion of LSD is being noticed by you, immediately report to your nearby hospital so that the implementation of control program should be instituted as early as possible, which will help in restriction of the further step of the disease. So that only you take home message with you. Thank you.
Manjunath?
Yes, sir. Like we are completed with the question and answer session, sir.
Right.
Go to Manjunath.
Okay. If there are no questions, sir, I have 2 questions to Sudhakar sir and 1 question to Manjunath Reddy. Okay. Thank you, sir. I will conclude with my 3 questions. 2 questions to Dr. Sudhakar sir and 1 question to Manjunath Reddy sir. Sir, Sudhakar sir, what about the characterization of the samples which have come positive from Karnataka state, sir? Has it been done, sir? Characterized?
Actually, that is official question. You can definitely discuss with me later on the telephone. Right? Hello, Molinath, I am not seeing you.
Disconnection.
Okay, disconnect.
Yes, sir. Because due to some problem, he might be having some problem network issues.
ಸರ್ ಅಲ್ಲಿ ಕಳ್ಸಬಹುದು, ಇಲ್ಲಿ ಬೇಕಾದ್ರೆ ಕಳಿಸಬಹುದು. ನಿವೇದಿಗೆ ಬೇಕಾದ್ರೆ ಡಾಕ್ಟರ್ ಬೇಕು ಅಂದ್ರೆ ಭರೋಸಾ ಇವರು ರವೀಂದ್ರ ಸರ್.
Okay. We'll send the samples. No issue.
ಮನೇನ ಕನ್ಕ್ಲೂಡ್ ಮಾಡ್ತೀರಾ?
ಇನ್ನೊಂದು ಕ್ವಶನ್ ಇದೆ ಅಂದ್ರಲ್ಲ ಅವರು.
Please unmute your mic, sir.
Okay.
Mine.
What are all the different TM lesions that you find in LSD, sir?
In PM lesion also, so far I have not done the PM and no mortality of any animal were recorded in Odisha outbreak. However, this nodular lesion you will find in the respiratory and digestive tract also. Moreover, in the severe case, in all the visceral organs, you will find the nodular lesion. And if you will take these PM samples and you will put to the test, that will give you the result.
Manjunath, sir, you want to?
Sir, it has been shown experimentally also. Experimentally infected cattle, once they sacrifice, and naturally infected also, all the epithelial, because it is epitheliotropic virus, wherever there is a type of epithelium meant for squamous nature or this one, and the pock lesions. Why placenta? Because if you see the chicken, sir, pox, even that's what placenta also you'll find in aborted cases, lesions of the pock. In the same way, throughout the GI tract, you'll find the lesions because it is epitheliotropic.
Okay.
Lungs allu siktave.
Okay. Immunity after in the recovered animal, sir?
Pardon? Recovered animal? So
yes, sir.
In case of natural or even vaccinated animal, immunity lasts for up to 1 year only, but it may extend based on the health status of the animal. Because humoral immunity doesn't play a major part in the in the LSD, CMI response is only predominant. So evaluation of the antibody after the vaccination or after the infection by virus neutralization test, because neutralization test will give you a result for only neutralizing antibody level. If neutralizing antibody level is there, then only virus neutralization test will give you the result. Otherwise, in recently IDVET has launched the ELISA. That ELISA also can detect the level below the neutralization state. And normally it has been reported that up to 1 year the immunity persists. So in this vaccinated cattle, annual booster is recommended for homologous LSD vaccine. In goatpox, similarly, we have to have some experimental trial. Then we can assess that means at what point of time that immunity vanishes and when the booster vaccine is required, particularly in case of India.
Thank you, sir. Thank you so much for your.
One slide, sir. Last in that it has been acted.
Yes, sir. Please, I go.
Yeah. In the last of my slide in prevention and control, I showed you one slide. Which we showed a graph, the seroconversion over number of vaccine doses given for the same animal. So at the first injection, you may not find the seroconversion what you expected. So by the third time you will find the exact required conversion. So minimum 3 times, in the sense 3 years, then the animal is going to be full immune. If you want that article, I share with you.
Okay, sir. Okay, sir. Thank you, sir. Thank you. Thank you for your all valuable answers to the questions raised by participant and including us. Sir, we have come to an end of this particular webinar session. So may I now request Mr. Shashi Kiran to present vote of thanks.
Good evening, everyone. Even though it's too late, but we need to keep up with the procedures. Myself, Shashi Kiran, student and president of IVSA Bidar. It's my immense pleasure to express the gratitude on behalf of Veterinary College Bidar, alumni, and IVSA Bidar for organizing this gracious event. So, my heartfelt thanks to the speakers who made the event a grand success. Firstly, I want to thank the speaker, Dr. S.B. Sudhakar, sir, who really gave a wonderful diagnostic approaches. Thank you so much, sir. Next, Dr. Manjunath Reddy, sir, who spoke on based on epidemiological aspects. Really, it was very knowledgeable session, sir. Thank you. So next, my heartfelt thanks I want to express Dr. Vivek Kasaralikar, sir, who gave us simple and effective methods for therapeutic approaches for lumpy skin disease. Even I extend my thanks to Dr. Dilip Kumar, sir, Dean, Veterinary College Bidar, who headed this event to make it successful. My sincere thanks to the professor and Dr. Manjunath, sir, who worked behind the screens to organize this webinar, and the person who collected all of us to enjoy this knowledgeable session. And lastly, the participants. Without you guys, it would be nothing. Thank you, participants. Hope you got some information and this made your day. Thank you, everyone. Thank you so much. We'll meet you tomorrow at 11 a.m. with another great speaker. Till then, take care.
Thank you, sir. Thank you, sir. Thank you, Sudhakar, sir.
Thank you. Thank you.
Thank you, Manjunath Reddy, sir.
Yeah.
Thank you, sir. Kasaralikar, sir. Thank you. Shall we end? Shashi Kiran.
Yes, sir.
Thank you, sir.
Thank you, sir.